and that would be a competition among all the participants who have given abstract. They are required to go above, and there they can compete. That is BP Chaudhary Memorial Award. BP Chaudhary was our teacher at Banibihar, a very renowned taxonomist. He has worked on ethnobotany also. And all this PC Panda, Professor PC Panda, Professor Kunjabihar Satpathy, and few others, they are direct students of Professor Chaudhary, who worked with him for PhD, for their PhD. So that will be held in the upstairs. And here there will be few invited lectures, and Professor Abidas, who is a very renowned scientist in taxonomy, biotechnologist, and Professor Ramlal from Ranchi University. They are the chairs, they are the chairperson and co-chairpersons. I request them to come over to the dais, Professor Ramlal and Professor Abidas. And now the session will begin. Ipsita will take over.
Good morning, everyone. I am Ipsita Priyadarshini Sammal, PhD Research Scholar from Department of Botany, Centurion University. Welcome to Day 2 of AMPRI 2026 and the 48th Annual Conference of the Odisha Botanical Society. I hope you all had a wonderful time yesterday and are looking forward to another enriching day of scientific deliberations. So we now proceed to technical session 2 along with the parallel session, that is BP Chaudhary Memorial Award. Now we will start the technical session 3. For technical session 3, we have Professor Anantabandhu Das, former professor and head Department of Botany, Utkal University, as our session chair, and Professor Ram Lal from Ranchi University as our co-chair. And I invite Dr. Madhusmita Barik, Assistant Professor, Department of Biotechnology, Centurion University, to as our rapporteur. Ma'am, please. Our first speaker of today is Dr. Shankar Narayan Sinha, Professor, Department of Botany, Utkal University of Kalyani, Kolkata.
हाँ उधर फूल आ गया तो ठीक है ओके Good morning everyone. At the outset, I am very much thankful to Professor Kunjo Bihari Satpathy and Organizing Secretary Professor Gyanranjan Mahalik for inviting me in this Odisha Botanical Congress and to use the podium for deliberation. Anyway, today my lecture will concentrate on some Hello. Now the invited lecture by Dr. Shankar Narayan Sinha. Phytochemicals as antimicrobes will deliver before the audience and its effect with relation to the advanced medicinal plants. Now I request Dr. Shankar Narayan Sinha will deliver the lecture on the appropriate topics.
Thank you, sir, for
And I would also like to place only, you have given only 20 minutes to finish your lecture within
Okay,
limit.
sir. Thank you for nice introduction. Now, the today topic is phytochemicals as antimicrobials. As we know that for the last few decades, public concerns has been voiced about the antimicrobial resistance to several chemical drugs or antibiotics. So, the emergence of this antimicrobial resistance has posed a serious problem, and it is a threat, menace to the public, and it raises question for the effectiveness of antibiotics, resulting in increased morbidity, mortality, and escalation of healthcare cost, causing a worldwide health crisis. Now, the overuse and misuse of antibiotics in global healthcare setup has aggravated the development and spread of antimicrobial resistance, leading to the emergence of multi-drug resistant microorganisms, which further restricts the treatment option and which creates a critical need to explore alternative approaches to abate the bacterial infections. Since the discovery rate of microbially derived antimicrobials is at its lowest since the golden era of antimicrobials, and the emergence of microbial resistance has further narrowed down the treatment option. So, exploration of alternative source of medicine based on plant products now become necessary. So, with this perspective in mind, so my discussions is to review the full possible spectrum of phytoconstituents discovered since 2000 till date and their potential to decrease bacterial resistance, and to discuss the activities of phytochemicals against bacteria with special emphasis on multi-drug resistant bacteria, possible combinations with traditional antibiotics, roles in current medicine and future perspectives. So, let us come to plant secondary metabolites as antimicrobials, and the most important, which is almost found in several plants, so that is alkaloids. You know that alkaloids are heterocyclic nitrogenous compounds and biosynthetically produced from amino acids and show variability in chemical structures. The activity of alkaloids against microbial infections is mainly attributed to their inhibitory actions against efflux pumps. And next, many alkaloid compounds have been reported to have marked significance in the treatment of microbial infections. Alkaloids found in the plant kingdom are structurally very diverse. However, irrespective of the diversification in the mechanism of action, plant alkaloids can be developed into potent antimicrobials that would not only revive the treatment options but also ensure the development of further resistance is prevented. Let us come to few alkaloids. So, I have mentioned few of the research on alkaloids. Here say berberine. You know that it is an isoquinoline alkaloid found in the bark of stem and roots of barberry species. An activity, antimicrobial activity against various microbes including bacteria, fungi, protozoa and viruses. And this is a study of Kamene et al. 2019. Then angerimine, it is also an isoquinoline alkaloid and you will find it pancreatum illyricum methanolic fractions, and it also showed significant antibacterial activity. Then piperine, there is a piperidine alkaloid from Piper nigrum, Piper longum, antimicrobial activity against Staphylococcus aureus and synergistically reduce the MIC values when administered along with fluoroquinolone antibiotics. And this is the study of Akil et al. Then maculin, cocosagin and dictamine, quinolone anti-alkaloids and found in stem bark of Teclia alphaceae. Significant antimicrobial activity. Then tomatidine and conacin, these are the steroidal alkaloid. You will find it tomato, brinjal, mainly Solanaceae family. Antimicrobial activity against Staphylococcus aureus, and this was a study of Gue et al. Reserpine, very common alkaloid as we read in our syllabus. Say this is the indole alkaloid from Rauwolfia serpentina. It inhibits efflux pumps and was reported to decrease the fluoroquinolone resistance in Stenophoma maltophylla. Then sanguinarine, initially isolated from Sanguinaria canadensis, later on Argemone mexicana, Chelidonia majus, Macleia cordata. So this plant, they showed antibacterial activity against methicillin resistant Staphylococcus aureus strains, Mycobacterium aurum and Mycobacterium smegmatis. This was a study of Newton et al. 2002. Caffeine, these are the xanthine alkaloid. So found in Coffea species, say Coffea arabica, Coffea robusta, Thea sinensis. They showed anti-quorum sensing activity against Pseudomonas aeruginosa. Then come to another phytoconstituent, that is phenols. Plant phenolics are recognized as important bioactive compounds, show wide range of pharmacological activities and strong pharmacological effects. Plant derived phenols can be found in simple or polymerized forms and contain an aromatic ring structure with one or more hydroxyl groups. Plant phenolics are categorized into many classes such as simple phenols, phenolic acids, quinones, flavonoids, tannins, etc. Say for example, catechol and pyrogallol, the simple phenol, they are allelochemicals synthesized by plants. They showed antimicrobial activity against Corynebacterium xerosis, not Corynebacterium diphtheria, xerosis, Pseudomonas putida, Pseudomonas pyocyania. Catechol was found to have an antifungal effect on Fusarium oxysporum and Penicillium italicum. Next, resorcinol. Source is Ainslia bonatii, effective against methicillin resistant, that is MRSA and ASBL, Staphylococcus aureus. Gallic acid and ferulic acid, very common phenolic compounds. Gallic acid in tea oak bark, sumac and fruits like grapes. Ferulic acid found in rice, oats and wheat bran as well as fruits and various vegetables against Escherichia coli, Staphylococcus aureus, Listeria monocytogenes, and Pseudomonas aeruginosa. Studied from Borges et al. 2013. Next, resveratrol, found in grape skins, berries, peanuts, Japanese knotweed. Activity against various bacterial strains, a multi-drug efflux system CME-ABC of Campylobacter jejuni and efflux pumps of Mycobacteria smegmatis. Then 3-para-transcoumaroyl 2-hydroxyquinic acid. These are complex phenols from Cedrus deodara, antibacterial activity against Staphylococcus aureus. You will find several antimicrobials, that is phytoconstituents, for abating the Staphylococcus aureus, which is one of the potent infectious agent of human body. Then cebulinic acid, as the name indicate, from Terminalia cebula, inhibit DNA gyrase of quinolone resistant Mycobacterium tuberculosis. Then quercetin and epigenin. So quercetin is most abundant in onions, apples, berries, tea, capers, while epigenin is found in high concentration in parsley, celery, chamomile and citrus fruits. They showed antibacterial activity against Helicobacter pylori, the dangerous infectious agent bacteria, and Escherichia coli. Catechins, another phenolic compounds found in Theasinensis, particularly green tea, bacterial cell membrane damage they do. Then bicaline, a flavone compound, Scutellaria bicalensis, Scutellaria lateriflora and Thymus vulgaris, they produce this type of phenols. They inhibit nor-efflux pumps, thus increasing the efficacy of antibiotics such as beta-lactam antibiotic, penicillin-like antibiotic, say ciprofloxacin, and tetracycline against methicillin-resistant Staphylococcus aureus. Then diacetyl A. Diacetyl A is a isoflavone compound found in Trifolium pratensis, then Cicer aritinum, Medicago sativa and soybeans. Inhibitory activity against MRSA and has been found to inhibit MRSA efflux pumps by study 2014 Zhao et al. Then camphorol, active flavonoid compound, tea, broccoli, grapes, tomatoes, berries, etc. Triazole resistant, active against triazole resistant Candida albicans and MRSA. Then epigallocatechin gallate, found in Theasinensis, that is Camellia sinensis. So it is very interesting to note the tea contains several compounds which is beneficial to health. They showed antimicrobial activity against MRSA by inhibiting nor-A efflux pump. Let us come to tannin. Tannins, trees like oak, chestnut and mimosa, as well as fruits such as grapes, persimmons and pomegranate. They showed antimicrobial activity on gram-positive bacteria, basically over gram-negative ones. These are the study of Tyagi et al. Also, we have studied this compound in our laboratory in 2014, that is 2014, that is printing mistake. Then curcumin, very common compound, isolated from Curcuma longa. They showed antimicrobial activity against Escherichia coli and Staphylococcus aureus. Let us come to another phytoconstituent, say terpenes. Terpenes are aromatic compounds found in many plants and are responsible for the characteristic smell of many plants such as cannabis, pine, lavender, as well as fresh orange peel. Apart from being primary structural components of cells, cholesterol and steroids in cellular membranes, they also act as functional molecules such as carotenoids, quinones and retinal. Say for example, say carvacrol, the essential oils of Thymus vulgaris, Lepidium flavum, Orantium bele and Bergamia and Origanum vulgare. So they showed anti-biofilm development activity against Staphylococcus aureus and Salmonella typhimurium and is reported to have activity against tobacco mosaic virus and cucumber mosaic virus, also effective against foodborne pathogens such as Escherichia coli, Salmonella and Bacillus cereus. These are the study of Alivi et al. And my laboratory study also Sinha et al. 2014 and Knowles et al. 2005. Thymol. Thymol is an essential oil component of Thymus vulgaris. Antibacterial activity effects on tetracycline resistant Salmonella typhimurium and Escherichia coli, penicillin resistant Staphylococcus aureus and erythromycin resistant Staphylococcus pyogenes. So study of Jadhav et al. 2015 and in our laboratory in the same year. Eugenol, you found in clove, cinnamon, basil, bay leaves, etc. Eugenol inhibits biofilm formation by MRSA and MSSA, clinical strains as well as the synthesis of virulence factors by Pseudomonas. This has also been studied in our laboratory. Ursolic acid. So this is the triterpene found in Mirabilis jalapa, many fruits and herbs used in daily life. Disorganizing effects on Escherichia coli, membrane and broad spectrum antibacterial activities. Eugenol and cinnamaldehyde, plant essential oils, activity against wide range of pathogens including Helicobacter pylori, causing damage to the cell membrane. Let us come to organosulfur compounds. Organosulfur compounds are sulfur containing organic molecules that are responsible for the strong aromas. Several organosulfur compounds such as allicin, azoine, dialkyl sulfides, S-allyl cysteine and isothiocyanates were found to be effective against both gram-positive as well as gram-negative bacteria. Say for example, allicin, diallyl thiosulfonate, Allium sativum, active against a variety of microbes including MRSA, Pseudomonas aeruginosa, Staphylococcus agalactium, Staphylococcus epidermidis and oral pathogens that can cause periodontitis. Azoine, it is also from Allium sativum by the study of Nakamoto et al. 2019, active against both gram-negative as well as gram-positive bacteria along with some fungal strains including Aspergillus niger and Candida albicans. Isothiocyanine, abundant in members of family Brassicaceae such as broccoli, cabbage, cauliflower and mustard, active against oral pathogens such as Helicobacter pylori due to high electrophilicity of an ITC carbon atom. It can react with amines, thiols and hydroxyl groups of cellular proteins. Various structures of the compound, I will not go into details of this. So let us come to the conclusion. Medicinal plants are still being used in domestic households for many infectious diseases. Despite the availability of modern antibiotics, many parts of the Himalayan region, particularly the tribal population, still practice and prefer herbal medicine over modern antibiotics. Although many species of medicinal plants have been investigated for their antimicrobial activities, extensive research is needed to explore the untapped reserve of phytochemicals produced by the medicinal plants. The phytochemicals could act as novel antimicrobials, antibiotic potentiators, or resistant breakers. However, the integration of plant-derived microbial agents into mainstream medicine faces several challenges, which include number one, the complexity of plant extracts, the difficulty in isolating specific bioactive compounds, and number three, the variability in compound composition depending on the extraction techniques and environmental factors. Additionally, there are hurdles in standardizing quality control and ensuring stability. So, future research should focus on the detailed understanding of phytocompounds, pharmacokinetics and pharmacodynamics, their interactions with antibiotics, and the mechanisms behind these interactions. Advanced biotechnological techniques such as genetic manipulation can be employed to enhance the production and efficacy of these bioactive compounds. Furthermore, modern analytical techniques and collaborative international efforts can drive the discovery and development of new and effective antimicrobial agents from medicinal plants, potentially offering novel solutions to combat antibiotic resistance. These are some references. Thank you for patience hearing.
Hello. Thank you very much. This is a very nice lecture by Professor Sinha with a broad range and of the phytochemicals which is from the plant sources against the antimicrobial activity. So, I request the participants, any questions, at least 1-2 questions we can take. Yeah, please. Professor Ramu Mahanti, sir.
Professor Sinha, we are very happy that you have given good account of secondary metabolites derived from microorganisms. I know you are a good microbiologist and you have worked so nicely in your lab. I have few queries. Like say, you uttered several times Staphylococcus aureus.
Yes.
This is very important pathogen which infects the upper respiratory tract mostly, and the alkaloids whichever is applied, when it is applied through Ayurveda, it works nicely when there will be suktam. Suktam is the liquid type of thing. It doesn't work in tablet form or powder form. That is one thing. Secondly, the divine physicians which are present profusely in our tribal community, they treat these diseases very nicely through their divine activities. That is to be highlighted here. Then secondly, about the Salmonella typhimurium, which you have said, unfortunately it's not a pathogen. Salmonella typhi is a pathogen. It causes typhoid. But Salmonella typhimurium is not at all pathogen. It's a very useful organism and it can be easily worked out in the lab for any kind of experiment. So these are the two things I would like to say, that your topic is very pertinent when the doctors alarm us that you should not take antibiotics at all. Whatever may be the case, antibiotics should be rejected. And in that case, your topic is very, very relevant. Thank you very much.
Thank you, sir, for this information.
Any other questions, please? Professor Chand, please. Because Professor Chand worked a long time in Taxol.
Well, not really. It's a curiosity out of ignorance. in your field of work. This is information I'd like to elicit from you. What are the standard tests performed in your laboratory to indicate that these different groups of secondary metabolites that you just referred have antimicrobial activity?
Sir, standard test is a normal Kirby-Bauer method, diffusion, diffusion or agar well diffusion method, and to find out the MIG value and MIC and MIB value. This is a standard method.
It's not MIG value, it is MIC.
MIC, that can be pronounced as MIC and nothing else. MIC, minimal inhibitory concentration.
I think he's talking about zone of inhibition.
Zone of, yes sir. By
These are all, these are all connected, zone of inhibition, MIC and MIB, it's fine. My second query would be, are these mechanisms almost similar mechanism of action of these different groups of these alkaloids, terpenoids, and sulfur-content groups? Are their mechanism different or almost similar against the microorganisms? The cell wall, does it affect the cell wall or something else?
Cell wall affected,
Yes.
in some cases 70S, 30S, ribosomal protein synthesis. In some cases, membrane damaging activities by inhibiting pumping system. It's a different types of mechanisms are there. Not all phytoconstituents show. Even in the case of antibiotics, different antibiotics show different activities. Similarly.
Actually,
Thank you, sir. I came halfway, but it was a nice talk, nice presentation. The phytochemicals that you cited, we have quite a good number of phytochemicals. Have you extracted them from fruits in your laboratory, or you have collected from different companies, Sigma or something like that?
Extracted.
Extracted. And purified.
From plant parts,
Okay.
leaf, flower, fruits, etc. From or whole plant. From plant parts that has been extracted in our laboratory.
actually, the mechanism, whatever Professor Chand was asking, I can add something that when we are talking about any of the phytochemicals, particularly on the bacteria, so it's a unicellular organism. So that's why it works fast in the membrane. Some of the cases, it may be in the translation level, it may be in transcription level, it may be in the what we call replication level. And also it binds sometimes with the enzyme system also. So that's why there are a lot of channels through which it can work, but specifically which chemical, no. That depends upon the particular chemicals and chemical structure. So that's why.
Even sir, sometimes what happens, it prevents formation of cell wall. So when there is no cell wall formation, we don't find the total organism. There is another process
And
also
some-
it takes.
There peptidoglycan synthesis, they inhibit. Cell wall.
And sometimes also it may be a bacteriostatic. No cell division is there, only elongation takes place. So it is also sometimes, number is fixed. So thank you very much. If there is no any other questions, Professor Sinha, and especially I thanks because of his poor eyesight. So that's why he has to take that computer. So thank you very much for this nice lecture and very good information. So any
Dr. Sinha, as you have mentioned here, the barberin, and what is the efficacy and what is the process of administration of the barberin?
Actually, these are the work of Barberin is not my work. Few works I have that is a review work of barberry is not my work. I have not done on
Actually,
barberry's plant.
actually this alkaloids, in which digit it acts and what is the process and what would be the doses? Can you
Actually, I have
explain
not
it?
mentioned here the mechanism of all the.
You work. Okay, thank you.
Just to give an insight of different types of phytochemicals and which.
Hmm.
not bacteria. Because that is the secondary goal. And usually we consider them as very important species because unless they are there, the organism cannot grow. It is a last one, policy.
Black-stem rust, Vaccinium graminis.
So that way, it cannot be considered anywhere relevant to bacterial inhibition. Thank you.
Okay. Thank you, Dr. Thank you for your nice lecture and very informative to the human beings. Thank you.
Now I request Professor Ramachandra Mohanty sir to felicitate Dr. Shankar Narayan Sinha sir. Next, we have Dr. Gatha Mohanty, Associate Professor, Periodontics and Oral Implantology, SOA University, Bhubaneswar. Ma'am, please.
Yeah, actually, this will be a very interesting topic because the last topic was the phytochemicals, and now actually in the real ground, a physical doctor can take the chemicals and then she will be evaluating us with the work that how these particular chemicals can affect the human to tackle the human disease. So, Dr. Mahanti, please.
A very good morning all, respected chairpersons, my senior dignitaries, and dear students. Yes, Dr. Sinha made my case very simple because he talked about the phytochemicals and there was a lot of discussion regarding what's the mechanism of action of these phytochemicals, whether it's alkaloids or phenols. So, like my topic is exploring the efficacy of the medicinal herbs. Now, me being a periodontist and an oral implantologist, you must be thinking or wondering why I'm here talking about herbs. But it is the efficacy of the medical herbs in the oral cavity because I deal with the soft tissues in the oral cavity, okay? So before proceeding into the main topic, I would like to make you understand, this is the contents. So what's a periodontium and what do I do? So if we consider a tree, so we have the shoot and the branches and the roots which lies beneath the soil, isn't it? Similar is a case of a tooth. So part of it is visible, that's the crown, and part of it lies beneath a structure which gives it a very good support, and that's the periodontium. And the periodontium is consisting of 4 parts, gingiva or the gums that you are able to see, the pink colored structure, isn't it, when you smile? That's the gingiva. Then comes the cementum, which gives a protection to the roots. Then is the ligament, which consists of lot of fibers which acts as a cushion hammock. Have you seen a hammock? When you have 2 trees and when you tie it with a hammock, you are able to sleep on it as well. So that kind of load can be burnt by this periodontal ligament. And finally, the alveolar bone, or the bony structure which supports all the teeth. That's why we are able to take up all the stresses, such as the vertical stresses. When we chew, you can also use open a soda bottle can, isn't it, with your teeth? People try to do that. So how is it able to take all the stresses, which is about 800 or 1000 newtons when you chew in the molars, and when it's the anterior tooth or the front tooth, it's about 200 newtons of pressure. It's all because of the periodontium, which provides it the support. Now, how many of you can say whether the tooth is living or non-living? I need the students to answer this. Is the tooth living? How many say yes? Everyone feels that it's a non-living? How many of you are in favor of yes? I want to see hands raised whether it's yes or no. I'm sorry, everyone says no except one. The tooth is living, of course, it has nerve supply. Some parts is non-living, but majority the tooth is vital. When we say a tooth is vital, how do you get the sensation? It's because it's living. We have the nerve supply, we have the artery supply. Similarly, how a tree is getting its supply? We get the minerals, you get the water from the soil. Similarly, everything is being supplied by the nerves and the vessels. So what's the burden of periodontal disease? Now, periodontium, when it is affected, is known as periodontal disease, and I deal with this kind of patients. The burden is that over 13 billion population in the world suffer from these kind of diseases. And not all patients are same. Now, to simplify the disease, it starts with gingivitis, that is inflammation of the gums. Any itis and a prefix to it means it's an inflammation, such as sinusitis. So inflammation of the sinuses, sinusitis. So gingiva starts with this, is known as the gingivitis, which progresses to a more aggressive form, which is known as the periodontitis, when the periodontium as a whole is affected. So slowly what happens, you will see there is redness around the gums, okay, there is bleeding, there is pain, and finally what happens is there is lot of recession. Recessions means the root is visible. Sometimes it happens with the plants as well, isn't it? The root is visible because the soil has receded down. The similar thing happens in the root as well. So how do I know that there is lot of bone loss? I have an instrument which is showing, do we have a pointer here? Do we have a pointer? So this is the instrument which is a marked instrument, which is, we tell this as a probe. So it has markings at different millimeters, like 50. This is a 15 millimeter probe. That means the probe has gone 15 millimeter into the periodontium. That means there is so much of destruction of the periodontium, which does not appear to us as that severe until unless we as periodontists see the case. Now this is a case of periodontal disease which has been treated. Even though there is a lot of recession, still the teeth are stable because they have been splintered well. Now why this disease is of importance to us? Because it is a multifactorial disease. It does not go alone. It takes a lot of factors around it. What would be the factors? I'll come to it. Now there is diverse microflora. The oral cavity has a maximum number of microflora. It could be gram-positive, gram-negative microorganisms, and it could be in state of symbiotic or sometimes turn parasitic when the normal conditions of a human being is associated with a comorbidity. Now, there are different stages of progression, and there is also a heredity which is associated with the disease. Now, these are the factors which will affect the gingival conditions. Okay, what are these factors? Let me come to it quickly one by one. One is stress. Any amount of stress, it produces cortisol, and that leads to the destruction of the fibers which are mainly consisting of the periodontal ligament fibers. Okay. Then we come to different kinds of drugs, such as we are taking antihistaminics, we are taking drugs for blood pressure, we are taking drugs for any disease, n number of diseases we having, neurodegenerative disorders, anything. So these drugs are chemicals, and these chemicals do have a role to play in the destruction of the gingival fibers as well as the periodontal ligament fibers in the long run. Okay. Then we talk about dry mouth, yes. Then certain hormones, like ladies have different hormonal imbalances which have a role to play in the destructions of the gingival fibers and the periodontal ligament fibers. This is one X-ray. Why I'm showing you X-ray? Because you will, now this is the second X-ray. Where do you think, which is a better X-ray? Better as in, as a doctor I'll see this is a better X-ray and this is not a good X-ray. 1 or 2? Number 1 or 2? Which is a better X-ray? Better as in number 1. Why do we say so? Because the amount of bone here, bone lies here, and see the destruction of bone here. The amount of destruction has happened here, and it is all because of the disease. Now, the major role of plaque and calculus is to create this disease. Plaque is nothing but a soft gelatinous matrix which is embedded with microorganisms. Lot of microorganisms which are commensals inside the oral cavity. Once they are embedded in this gelatinous matrix, which forms an oral biofilm. Biofilm, as you know, when you allow water to stand on a bucket for a few days, you have a soft slimy layer. Similar thing gets deposited on the oral cavity. But what do? We brush daily. So that by brushing or by mouth washing, this plaque washes off. But if you do not brush properly, then this plaque, it mineralizes to form the calculus. Now this is a mineralized form of plaque. So this calculus destroys the periodontal ligament structures. So this is, I'm not going into this because I have to cover a lot of slides. Now this is a normal pocket. This is the periodontal pocket, where there is a bone loss, there is loss of attachment, and this is where exactly the microorganisms act. And they are mostly the gram-negative microorganisms, which are aggregative-bacter actinomycetum comitans and Porphyromonas gingivalis. They are the keystone pathogens causing destruction of the periodontium. So our job is to seal those pockets and prevent the progression of the disease. Now, when you saw my topic, it was with morbidity, without comorbidities. Now, what are the comorbidities? These are the factors: hypertension, blood pressure, as in you say, okay, diabetes, any kind of chronic kidney disease, cardiovascular disease, and these are all linked to the oral cavity, that is the periodontium, which supports the tooth structure. Alzheimer's disease is now linked to periodontitis. Diabetes is the 6th complication of periodontitis. Obesity, pregnancy. Pregnancy, we have low-term birth weight babies because of periodontal disease. So everything is now linked. So we have to be very careful while we are dealing with patients with periodontitis. Now, how do we treat this disease? We normally go for non-surgical treatment and surgical treatment. If it's a non-surgical treatment, we have lots of options. We do manual instrumentation, which is known as scaling and root planing. We do diet modifications in diabetic patients. We do splinting if a tooth is mobile. we use lasers and we do a local drug delivery. That's known as the local drug delivery. I was talking about sealing the pockets. How do we seal the pockets? We cannot suture it if the patient is with a comorbidity, because the patients who are diabetics, the patients who are hypertensives, the patients who have cancers cannot be treated surgically. There are some contraindications. We cannot treat these patients surgically. So what do we do then? There comes the role of the medicinal herbs. So we know that, does the disease progress? Yes, because the patients are not always the same. They do not listen to us. Their systemic health could be a factor. Their age could be a factor because healing is compromised there. There are a lot of environmental factors such as smoking, stress, and there is non-cooperation from the patient. And also genetic susceptibility. Genetic heredity has a role to play in aggravation of the disease. So what's the case? Normal treatment protocol for a no morbidity patient, like the patient does not have diabetes, the patient is just a periodontitis patient. We can treat him with non-surgical as well as surgical way. We can do surgeries, we will seal the pocket, we'll close the pocket, and absolutely the patient is fine. But if the patient is a morbid patient, comorbid patient, then what do? We assess the stages, we assess their grades, and then we discuss it with the patients that we cannot perform the routine treatment which is available for the patients without morbidity. So what we have to do is do a local drug delivery, because we have to look for targeted treatments into the pockets. We cannot give an oral drug, we have to target it inside the pocket, and what better than the plants, which are much safer, do not do any harm, as Dr. Sena has already discussed. So it has reduced side effects and also enhanced efficacy, because 100-fold presentation inside the pocket will kill all the periodontal pathogens. So they are given in forms of fibers, strips and films, microparticles and gels. So we have irrigating systems, and then we have strips and gel system. The periodontal disease market, because as I already said, there is lot of scope for this kind of research, where plant products can be used in different kinds of formulations and brought out into the market after patency, and patients can be used, because there are 13 billion people out there with periodontitis and oral diseases. So why medicinal plants? Because Ayurveda has been using a lot of plants since ages. There is a lot of experimentation on these plants, and they consider that there has to be an equilibrium between the 3 biological humors, that is the doshas, the 7 body tissues, that is the dhatus, proper digestion, and the state of pleasure or happiness of the soul exists. For oral hygiene, oil pulling has been used. Oil pulling is something which is known as gandusa, where oil is taken inside the mouth and you swish it for some time. It has an antimicrobial effect, as well as saponification takes place. Saponification is formation of foam. Okay? Oil, have you ever thought that oil should be used as a mouthwash? Yes. Once you swish it for some time and spit it out, that oil becomes totally liquid and it creates a foam, which has a cleaning efficacy, and what we clean is, remove is the plaque and the calculus. Similarly, Jiva Lekhana and Dant Dhwani, that is chewing of herbal brushes. You must be knowing people using karanja, the neem tree barks, and also guava barks have been used as toothbrushes in the past. Similarly, neem stick, fresh stems of licorice, black katochu or the kach tree, arjuna tree, fever nut, milkweed plants have been used by the Ayurvedic system for brushing. Tissue regeneration is closing of the pocket, where we try to regenerate the lost periodontal structures by either surgical methods or by use of local drug deliveries. Okay? So a lot of forms have been used, such as amla has been used, licorice has been used, bilberry and hawthorn berry fruits have been used. Now these 2 slides, I'll be showing what are the different kinds of herbs which are used in the periodontal disease. And the main aim of these herbs and the chemicals is to fight the dental plaque, which is the main agent that causes the disease. Amla, vitamin C, antioxidants and the tannins. It has got an antioxidant effects, and it works on gingival inflammation, aphthous stomatitis and mouth ulcers. The anar or the pomegranate, it has been used, the seeds have been used, the pomegranate as the bark has been used, the fruit, the cover of the fruit is being used. All these are being used to reduce the gingival inflammation, antibacterial and antifungal activity. Similarly, the clove, you have heard about the clove. Whenever there is a toothache, people normally use clove oil, isn't it? That clove is being used, and the active components is the eugenol and the flavonoids, and that also has an anti-inflammatory and analgesic property, and it's also used in the root canal therapy, which is quite common these days, where we make the tooth non-vital. The tooth which was vital is having some pain, so you remove the nerve and have the root canal therapy to make it non-vital so that it is functional and it stays inside the oral cavity instead of removing the tooth as a whole, which was done in the past. Okay. Then we have Dati 1. I'm not going everything into details. Neem, Nilgiri, Sesame, Triphala. So this is from one of my review articles. I have taken this. Mostly they have the anti-inflammatory properties and the anti-plaque properties. Okay. This is another case. We have turmeric, we have curry leaf, we have tea tree oil, we have wheatgrass, licorice root, meswak, barberry, myrrh oil, green tea, shiitake mushroom, tulsi. This is a long list. I cannot go on and on. So there is a lot of scope for you students to take to research on these products. Now, what are the formulations? Once we identify the phytochemicals, as Sena sir has already said, that these phytochemicals from these plant products are useful, then what are the formulations that we are going to take? It could be gels, it could be mouthwashes, it could be targeted therapies, liquids into the pockets, or it could be healing packs. While after surgery we can give these packs which will help in healing, or it could be suspended release patches which can be placed in different oral mucosal lesions, okay? Or the nanoparticles. Now coming to the mouthwash, normally we have seen the mouthwashes when we go to the counter, we ask for a chlorhexidine mouthwash, a hexidine mouthwash. Commonly आप कौन सा mouthwash ढूंढते हो? What you see in the over the counter or you see in the televisions, right? Isn't it? We ask for that kind of mouthwash. Now, have you ever thought, how does a mouthwash work? I'll tell you how it works. Now, the tooth, you have a plaque which gets deposited on the surface of the tooth, okay? Now, the normal mouthwash is a dicationic molecule. It has two positive cations. One cation, it gets attached to the surface of the tooth, okay? The other cation it's like a pin. So when the So when the bacteria comes closer, the pin will go and puncture the cell wall. So that's how it acts. So I have to now once the herbs are identified, then we have to take the herbs, we have to dry them, we have to make any formulation and find out the minimum inhibitory capacity and the bacterial concentration at which it can be used, and then we find out the total antioxidant capacity, take the trial to the animals, and then we have the randomized control trials on the human beings so that we take it to the market and patent the drug and bring out in the market for use. So these are the studies. You have the microbiological gel agar diffusion culture studies. We have the animal studies, but everything has to do with ethical, so we have to take ethical permission for the animal studies and for the human studies. And then this is a split mouth, this one. So a single individual will be given 4 types of drug and it will be tried on. And to conclude, herbal extracts are being used in periodontal therapy to reduce gingival inflammation as anti-plaque agents, as antiseptics, antioxidants, antimicrobials, antifungals, antibacterials, antivirals, and analgesics. But lot of studies needs to be done in order to understand these phytochemicals, its action on the tooth and its action on the dental plaque and the microorganisms which act inside the mouth. Thank you so much for a patient hearing.
Thank you very much, Dr.
These are my
Mohanty.
references.
Now the session is open for discussion. Anybody can ask any query, any type of questions.
I can Actually, everybody has their kit, so everybody
Ah,
can
ah.
ask the question.
Actually, actually the speech is very much clear and nice one. However, anybody can in class. Okay.
Theme, so addressed to the theme very well. Well, apart from that, from her own experience, I'd like to get her report on evaluation of the different types of formulations which you have already made yourself or commercially available in market. So what is your experience of evaluation? Which one of these herbs would you recommend for a specific case? If you have, I know you have already tested them.
Of gels and mouthwashes.
Yes.
So normally, depends on what we are using on. So if it's a mouthwash, like we have an antibacterial mouthwash or antifungal mouthwash, or if it's a lesion, then we have patches which can be placed on the top of the lesion. So suspended release. Slowly, the patch will release the medication on the whatever the ulcer is there or something is there. So that's how we do it. And before that, we have to do these studies, whether it's acting or not. You cannot say that the phytochemical has been identified. These are the properties of the phytochemical and it is going to act on that. But we have to do certain studies which are known as the randomized controlled trials, where we try the gold standard which is already there and established on the patient, and the new formulation in another group of patients with similar kind of diseases, so as to claim that yes, our drug which is better, it has a better efficacy or similar efficacy to the gold standard.
And do you believe that different phytochemicals available in different medicinal herbs that you already tried have a synergistic effect? Or you can just try with one of the purified chemicals from one particular herb or one particular plant and try it to be more efficient?
It's one particular plant. We haven't used any synergistic effects, but it's one of the plants. Just such as aloe vera. We have used aloe vera, and we have used neem also.
You have
And
used
we
them
have used,
separately?
yes, all separated. Myrrh also, myrrh oil has been used. And also the guava extracts and a lot of cranberry, a lot of extracts have been used, but individually they have been used, yes.
Right. Thank you. It's very, very interesting. I really
Thank
enjoyed your lecture.
Thank you.
Ma'am, I didn't see one plant. It's very common in Odisha. This is what I would call. That's Melotus. I didn't find that plant. It can be the very useful. In
Sure. I'll take that into
Yes.
consideration, sir.
Another query regarding the Triphala. I'm using in the early morning, means empty stomach. I'm taking that Triphala in the means.
Yes,
Putting in the water and that it's a rinsing of the mouth.
correct.
This is what I have heard, that it is the reducing the plaque formation.
Plaque formation, yes,
So
yes.
whether is it true, that means it is a very useful. in
Yes,
that?
yes, absolutely. Absolutely, it's
Okay.
true.
If somebody will want to use as a means tooth powder, means the person can. Put
No,
in the
powder is not advised usually
Okay.
because it leads to a lot of aberration of the tooth. So
Okay.
we do not advise powder. It's a mouthwash or a gel formulation of a
Okay.
toothpaste.
Yeah. Okay. Thank you.
Hello. Actually, this Melotus philippensis, Sinduri, it is not usually used by the people of Odisha as this toothbrush stick. But two plants you have not mentioned, whether you have come across this literature, that one ritual is there in Lord Jagannath.
Okay.
So Lord Jagannath is offer this mumus of LNG, that is Bauda, and
Bauda.
Mutturi, that is Smilax jalanica. So these two, yes, these two sticks are given to offer to Lord Jagannath before breakfast. So can you try?
I think I should have to try that.
Nodiflorus, actually it is not used normally. Okay, thank you.
Ma'am, you will be happy. that my PhD's first PhD scholar is working on periodontal disease,
Oh, good.
and already we have the nano formulation, we have done the animal studies, and before the going to the, you have done the randomized studies, we have done a lot of computational tools, and this plant is totally a herb and new to the thing, and later on I am going for the nanofibers, but the gels are showing great results.
Yes, they had come for the ethical, I know that.
Yeah, they have
Yes.
come for the ethical committee report, and this is my
Yes,
first
yes,
PhD
I know
study.
that. Yes.
Kalpita has done wonderful thing that she has made some formulations, and you try your formulation.
Yes,
because
that
she
has to be tried.
She has tried formulation, and now she has got the UK patent. So
Okay. Good, nice to know that.
good.
Okay,
You should try that.
Yeah, thank you very much, because I know there will be a lot of questions, because everybody has the teeth, but we have the time. So, yeah, I have actually just convey this information that I have also plugged my teeth with madam. So she is a very nice doctor, at least I could say, as well as human being. And one more thing I just want to know, that lot of peoples may be interesting for that. When you are going for tooth bleaching, so is there any connection with this particular disease?
No.
No. So this
That
is safe.
on the
This
tooth
is safe.
surface. This is on the tooth.
So this is safe process. So anybody can go for, because for the beauty purpose, a lot of
Yes,
people
yes,
are going for that one. Okay, thank you very much, and with a big round of applause. So thank you very much, thank you very much. And one more information, actually, Professor Mahanti's daughter is Gatha Mahanti, and Professor Mahanti has very tremendous experience and expertise in plant science as well as microbiology, and rightly put into a daughter to experiment this one in the physical condition. Thank you very much. Like father, like daughter. Thank you very much. Thank you very much. Can go for the felicitation. Madam, please.
Now I request Professor Dr. Kunja Bihari Satpathy sir and our guests to felicitate Dr. Gatha Mohanty ma'am. Next, we have Dr. Gopal Krishna sir, Scientist C, Botanical Survey of India, Kolkata. Sir, please share your insights with us.
Yeah, Gopalji is from BSI, so he will be stamping on the plant whatever we are discussing with regard to the medicinal plant. So please, Gopalji, please.
Working? Okay, fine. It is great privilege and honor to present my experience of past in nearly around 20 years in front of the botanical fraternity, research scholar and the students. I am really very thankful to the organizer for invitation to deliver the talk on this auspicious day. Specifically, Professor Kunj Bihari Satpathy sir, who is the president of OBS, and Dr. Gyan Ranjan Mahalik, organizing secretary, OBS. Gyan Ranjan's, when I heard his name, it reminded me the para homonym. One of my embassy friends' name was the Gyan Ranjan, but his name was the Gyan Ranjan. But sir is Gyan Ranjan Mahalik. So thank you, sir. Basically, usually whenever I am delivering the talk, mostly it is related to the student, means I'm focusing that the students should get benefit of that. particularly the undergraduate and the postgraduate, and few of the students among the gatherings, they know me in person, that generally I deliver talk on the botanical nomenclature, so that person can understand how a name is originated and how a person can make them Latinized. Basically, this is some of the things what I was delivering in the past. And also I delivered talk on the plant identification from the ranunculus to the poaceae. So how this evolution is come in between, and how students can take it so that they can easily identify that. So this identification tool I discussed. But here this is the entire different talk. It's neither the nomenclature nor the plant taxonomy pure. So it's I would say that's in between, the transition between these two, where I have noticed during the last 20 years. This is what I am going to discuss here. So the topic is evident that it's the role of plant taxonomy in the ethnobotanical studies and some interesting facts. So what I would say, ethnobotany, we all are the ethnobotanist. Only thing, we have to identify our potential, what we can deliver to our society. Next slide, please. Oh, okay. Right. So ethno means the ethnic, referred to people, culture, a culture collective body of the belief, aesthetic language, knowledge and practice. This is what the definition we can see, even the AI tool will suggest to us. Ethnobotanical knowledge encompasses both wild and domesticated species. This is quite interesting. Whenever we are thinking of the ethnobotanical study, this is not only includes the wild species. This can be the cultivated species as well, and it rooted in observation, relationship, need and traditional ways of the knowing. One of the very great ethnobotanist, if we are knowing, this is from the Father Horsworth most possibly, when he came to the India and he wasn't able to find one of the disease, that person got married to a tribal woman so that they can find what they are using for the contraceptives. So such kind of things, it comes under the observation, which makes the person very popular by their research. So likewise, such knowledge evolves over time. It's not a one-day job. It needs time. So once we will spend the time, we will get the information and the knowledge as well. So this is a very popular picture. How many of you know this picture? Anyone? Raise your hand. Okay. I'm telling something in story. If you will see in picture, out of that only two are the Indian. His name is the Masi Sandhya and the Vedivel Gopal. These two persons are from the Andhra Pradesh. In 2005, in Florida, you know the Florida is in the USA. So what happened? In the zoo, there is one Burmese python have been escaped. Or because their population were more, so they made it freed. So what happened over the period, they find that the zoo authority, many of the avians, many of the mammals, many of the reptiles, many of the birds is gone missing. So they were not able to find out the reason what is going on. So later on, when they find that there is a snake, that's a python, it's nothing, it's the Burmese python, the originating from the India, that has been released and they are eating all the avians and birds. So now the problem for the entire Florida scientists to catch the snake, they spent thousands or lakhs of the dollars. Even their zoologists, their botanists, their agriculturists, they all come together on a common platform and starting searching this python. They were not able to find out the answer. So later on, means in 2017, I'm talking after 12 years, one young agriculture botanist came and he said, I need the $5,000. Everybody were wondering, what you will do of that money, gentleman? He said, nothing, just I want a ticket for the India. He came to the Andhra Pradesh with the help of some of the local people. He hired these two gentlemen, this name Masi Sadhya and the Vedivel Gopal. Within 7 days, that person catch nearly around the 30 pythons. Just you imagine, America is a superpower, they have the technology, they have the everything, and they were not able to find the python, but our snake catcher, within 7 days, they catch them, because they were knowing the hiding place. And the origin of the snake is from India, and they have the better understanding than the technology. So such kind of information should be spread among our young generation. Though it's a very low cost kind of job, nobody, even if I'm a snack catcher, I want to be say that my kids should be follow the same trend. It's not a kind of job what we are promoting, but such kind of things should be promoted so that this information will be go on one generation to the other generation. This is one of the example what a ethnobotanist can do. I'm sharing the experience what I have seen 2006 to 2010. I worked in the Odisha for these districts, Sundargarh, Fulbani, Mayurbhanj, and Angul. These 4 districts I have studied, and there I find some of the interesting facts, like what we do in the field. That first thing, we found some of the tribal person or any local person. We are asking them, we are recording their voice, because if I am not knowing the language, we should record that, so that later on somebody can help you in and you will get what they are saying. Another, if somebody is knowing your language, then you can write on the notepad. And if somebody is not going into the forest, then you can visit the local people. So these are the method of the data collections. Now, herein, I will share the information on the edible plants. This is very common in the Odisha. If you will visit the local market, you will find the mushrooms of plenty basically in the rainy season. You will find plenty of the kind of mushrooms in the Odisha is found in the market. And these are some of the species, the Terminomyces hemai. Another species, Terminomyces microcarpus. These 2 species very common. Is the correct identification is important, even if we traditionally recognize the species? This is a question to us. अभी हम लोगों ने मशरूम देखा, 2 mushrooms were edible. We will go into market and we will buy. Okay, fine. Now question comes, really traditional mushroom proper identification is very important. You look into the case. This photographs is from the Neora Valley National Park, West Bengal. One of my helping hand, he has taken all the mushrooms from there. And in the afternoon, we have this kind of delicious mushroom. Same thing happened in the Neora Valley. This story, if you will remember, then you will think about that. Yes, the correct identification is very important. What happened? These local people, they have collected the similar kind of mushroom which looks very similar to each other. But season was the different. So what happened? Once they collected, they came to the house and they wanted to make this mushroom. But what happened? Luckily, some person has invited that local person for the marriage ceremony. So that person didn't have that mushroom. They spare with their neighborhood. So in the later or the next day, almost 7-8 people killed by that mushroom. Even that person was knowing that this mushroom is the edible, but that was not the actual mushroom because the season was the different. So whenever we are having the ethnobotanical information, the correct identification play a very significant role, and we should know what is the correct mushroom or the correct plant. This is the one example. Look at this case. Here in tender leaf of the Bahinia, Purpurea or the Racemosa. This is very common in the Odisha. Now question to us, what is the species name? If you will go into the literature, you will find that both species is mentioned there. But what species in actual? If you will see the distribution of the species, most of the places you will find the Bahinia racemosa. So in my opinion, the species will be the Bahinia racemosa. It's not the Purpurea, it is the racemosa. Another species, the Indigofera cassoides. This is a tree species. You will find they are selling in the market. They are edible and very nutritious as well. Look at this example. This is from the Simlipal Biosphere Reserve in the Gurgudia range. This is from the Gurgudia. That person having the some cut is taking the latex from there and applying on the wound. I don't know how much effective, but that person was doing. So I believe that this information is true, because in person I have seen that species was used by that tribal person. Now question comes again. If you will see the leaf, very few can identify what is the actual species. Both species looks very similar. But the correct identity is the here, Calotropis gigantea. So this is the what I wanted to say. The role of taxonomy is here, the correct identification.
Which one is medicinal mostly?
Sir, this is gigantea, this is what I do. Yes, sir. I'm going back or sir back? Oh, okay, got it. This is the back. Now question comes, role of rank of taxa in disease treatment. We know this in early 1981, HIV or AIDS means spread in the entire world, and the people were more worried about that. So during that time, a group of the botanists from the America, they started searching some of the herbal plants so that they can cure the HIV. Some of the plants having the potential, and the plant was from the Indonesia. So a group of botanists visited the Indonesia. They went there in the place, the details was from the herbarium, and collected the specimen, go for the chemical screening. They didn't find the same character what plant was having in the herbarium, but the same species looks similar, but was not having the same potential. So all become very worried, what is going on? So later on, a person who was the expert of that group, that plant belongs to the Clusiaceae, so he went to that person and meet to that botanist. Then he find that the species what you are searching, it is not the species, it is something different. It's a new variety in fact. And later on, the happy ending was that the plant was found in the Singapore Botanical Garden, which have been taken from the Indonesia in long back. What happened? The local people were not knowing what is the actual identity of the species, and they were used as a firewood. So in that firewood, all germplasm have been destroyed. So some of the time, here in the case is the plant looks very similar, but the specific epithet is ranked at the variety was not having the same potential as the species having the potential. So sometimes this also happens. Some unique medicinal garden in Odisha, if you can see that in the Ramtirtha, long back I have taken this photograph. And there is a human body, so according to educate the people, each place having the one plant they have given, so that can be very useful for the students as well. Some of the articles we have the published. This is the sal teeth stick that can be used. I think Madam has also added this plant's name. And this is the use as a rope, the Buchnania, Cryptolepis buchnani. And yeah, this is what I have seen. A girl was collecting this plant. Then I asked the question that she said that I am using for the hair growth. That's true. It's nothing new. Yeah. Now some of the burning question of the ethnobotany. These are some of the questions. What are the people conception of the plants? What use is made of plant for food, medicine, material culture and ceremonial purpose? What is the extent of knowledge of the plant? In what category are plant names, word that deals with the plant group in the language? What can we learn by the studying this? So many questions are there. Now I'm moving to the another state. This is the state means Arunachal Pradesh where the sun rise first. In year 2011, I have the one big expedition. If we are just crossing the river Dibrugarh, then easily you can reach to this thing guest house. Beautiful guest house. After having the guest house, means beautiful, spend overnight. Next day we visited the local place, and they find in the market the two species was Disoxylum excelsum and the Iliocarpus floribundum was the selling in the market. This is very common in the Arunachal Pradesh. So this is the actual species. This is what they are edible in means selling in the market. But when we went to the forest, means Upper Siang district, there's something interesting I found. That was the role of the plant in killing the musk deer. We know the wealth of the means how much what is the value of the musk And if a plant can be the reason for the killing the musk, it is quite a very sad part of the ethnobotanical study. Herein what happened, there is a species name, I don't know exact species, the genus name is the Hymenidium from the Apiaceae. What is happening, this musk deer, so innocent, every early morning, they used to come and have the used as a fodder. The hunters, they are hiding somewhere else and just waiting for the early morning. It's around 3 to 4 a.m. early morning. Once they started having the fodder of the Hymenidium, and they shooting the musk deer. Illegal selling of this musk, they are coming from the Arunachal, then from Arunachal to transfer to the China. This is happening. So this is the another sad part of the ethnobotanical study. Even a person, local person knowing this is the endemic species or this is a very rare species, rare deer, they have the right to survive, to get the sugar. For a basic needs, they have to travel to get the sugar. For a basic needs, they have to travel around the 50 kilometers, 60 kilometers on the hill track. Even the post-independence, we are not reaching up to that level. So this place, if you will see that in this picture, there is a post office, there is a circle office, there is a helipad, everything is there. Nearly around 36 people are having the entire village. 36 houses are there. There is a one hydroelectric power. From there, in the evening hour, 3 hours they are getting the current. And if you will see their lifestyle, it is a far excellent than us. Much more happier than us, in fact. That lady is from the Adi tribe. If you are knowing if you will just Google it, and with the help of the bamboo sticks, they were cutting the umbilical cord. Because of that infections, most of the mothers was going to die. This was the most important, this was the only reason because of their population depleted, and nearly around they have surveyed less than 100 or less than that means survived in as of now. So what is the beauty of that house? If you will visit any Adi house, they will keep the utensil on the very proper way, clean and neat and clean. If you enter into the house, they have the automatic gate with the help of the lock and key. There is no lock and key. You enter, gate will be automatically locked. The kind of hinge they are making from the bamboo sticks. If you want, you can have the food, there is no problem, but you have to keep the utensil and everything in the same way. This is a plant, this is called the Litsia cubeba. Most possibly this plant is also found in the Odisha, not a big deal. It should be, in fact, I believe that. Specifically in the Simlipal Biosphere Reserve, this should be there. Actually, cubeba means having some citric flavor. If you have visiting some of the restaurant, many of they are using this cubeba for adding the flavors, but very expensive because it's not a very easy plant that you will get everywhere. So the people, those who want to earn, they can also use for their sustainable development. I'm saying that this kind of plant they can plant everywhere. Now, exploitation of the plant, why these plants are getting exploited, the possible reasons are here in lack of the knowledge, education, motivator, no proper training. As I said in earlier slide, that Hymenidium, that plant is the fodder for the musk deer, but the musk deer never knows. Look at some of the example. Mendigofera colescens. If you will visit the northeast India, the local people have the two kind of the observation. First observation, a plant, if their fruits are inside the soil, that is having the medicinal property. Second one, a plant having the latex, that is the very highly medicinal plant. So two concepts are there. Just you look at the case of this Mendigofera colescens. This is an subalpine and alpine, in between this plant species is growing. This is belonging to the Solanaceae plant. You see that here in this fruits is going into the inside the soil. Because of that, they are exploiting this plant tremendously. And you will, if you will visit in the next season, you won't find this plant is happening there. So this is another problem. Another plant which is highly exploited in the Arunachal Pradesh is the Podophyllum hexandrum. You search in the Google, this Chinese people has made a very great mistake. They're doing the research on some different plant, But in place of that, this plant is the scapegoat of that. So all people are exploiting this plant in place of the actual species what it is in the China. This species is not happening, not growing in the China. But similar looking, having the species, the identity is a problem. Because of that, this species is highly exploited. If you will visit the Arunachal Pradesh houses, you will find a very big kitchen. And inside the kitchen, you will find the place where you can sleep if you are not in the from that place. And they will have the they will welcome you very perfectly. This person was discussing about these two plants, means the Podophyllum hexandrum and another species, that was the Mandigora pubescens. So these two species, that person was asking to our botanist, Dr. M.K. Pathak sir, and during that we have come to know about these plants. So whenever you are staying with the local people, you will get the information. In 2008 to till date, now moving to the some different part where I work, this Brucia mollis, if you will Google, you will find that this species is highly effective in the malaria, and that's true also. This species is the very limited, and I have only seen in the Buxa tiger reserve. Another species, both looks very similar. If the plant will dry, no one can identify which species is which one. Very difficult. But here in, the identification is very important. The species, the first case, if you see that, Phlogacanthus thersiiflorus, this species is the edible, the very tasty. The inflorescence is the cut and they are making the vegetable, but that species is not useful. But for the other person, if they will dry, that will be looks very similar. Now in coming to the Neora Valley, this is the forest staff, and when I stayed with him, that person said, "Sir, आज हम आपको ना आयुर्वेदिक दवाई खिलाएंगे." What is that? He said, if you will eat each and every day, the blood pressure will be lowered down. I was wondering what plant they will say. When he started plugging this, you know, sir, this is the nettle sting plant. Nettle sting plant you cannot touch with your bare hand. Once you touch means you are in the problem. It will be itching you, irritate you. So this nettle sting plant they cut down. I was wondering, this is the plenty, and this is one of the noxious weed in the Neora Valley National Park. Everywhere this species will be grown up. So what he did, after cutting, now you can see in his hand there is one stick. With the stick they have the putting on the, means forcing over the rock surface, and all the nettle hairs gone. After that he boiled in the water, and the taste was very much similar to the spinach. Really it was very delicious. I have already experienced the another species of that, Electostema, in the Arunachal Pradesh. So then I found all the urticaceae member having the nutraceutical value. If you will eat, you will find that yes, they having that potential. Another species, this was the actual species, nettle sting, Giardina diversifolia. Another species, how the ethnobotanical information is very useful in the identification of the plant. You know, when I saw this plant for the first time, I wasn't knowing what is this species. I was wondering what it could be. I was just discussing with my friends. Then I found that is the red berries. When I touched this red berries, I saw that. Then I taken out the plant from there. Then we will found the fasciculate roots. This fasciculate roots is the only found in only one plant of the Gomphostema. That species, Gomphostema mastercii. Master was one botanist named after the Hooker has made this species named after him. Then because of this red berries, I could recognize, okay, this species is nothing, it's a Gomphostema, because the Gomphostema having certain property, and because of that I could recognize this species. Here the case is the different. Because with the help of ethnobotanical information, I could identify the species. And you see this color, how it is coming out. Another species is very common, but it is very useful, that is the Calamus erectus in the Neora Valley National Park, which is useful in the control the blood sugar. That's true. Some of the species is used as the sauces. You can use it is very common everywhere. Persicaria. Another species, this is the member of the Ericaceae. If you can see that, there is one birds, they are taking the nectar from there. This is the Agapetis saligna. Very similar another species, Agapetis serpens. In the forest, while I was surveying, my forest staff said that, sir, muri khavo. हम muri कैसे खाएंगे? तो sir, we don't have onion, but we have that saligna. So he make all the slices of that. It was very delicious. If you will visit the Arunachal Pradesh, if you are visiting the Sikkim, you will find that rhododendron juice. So because of the same group of the plant having the tendency, having the some kind of flavor, because of that this plant is used for that one. Now, what we are missing? The carpological collection means the collection of the fruit making in a boxes, and they are putting the that can be used by the done by all the college students. Whenever we are collecting fruits, hardly we are putting any effort. But our request to you all, don't give it should not be like that. You have to put in your box, and it can be used as a serve for the carpological collections. कुछ नहीं है sir. We know that this Paplu Puspangadan sir, very popular personality in India if you are searching on the Google one, and he's no more among us. He died on the 19th December 2025. So this plant is dedicated to him because this plant make him the man of the tribals in the Kerala, and lots of development has been done in this Trichopus zillenica. When I visited the Kerala, during that time, only one question to this forest staff: can I get this Trichopus zillenicus? That plant I brought to my garden, I planted, and that I saw in the flowering. I am very happy that, and this plant is dedicated to the Puspangadan sir. Thank you so much. Please do ask the question, and for the students, I have the email ID. Somebody has asked me about the etymology and all, so do ask the question, I will answer you all. Thank you.
Only 1-2 questions we can take.
Sir, please sir.
Otherwise, we will be having a tea break, and during that time also we can have a discussion.
Sure, sure.
Thank you very much. Very, very nice and illuminating talk. Because I was also in Arunachal Pradesh for 2 years.
Yes, sir.
I know a lot of facts, whatever you are talking about.
True, sir.
Okay, thank you very much.
Thank you, sir. We now break for tea. Please be back in 10 minutes for oral presentation. Oral presentation will continue thereafter at the same venue here. Thank you.
दीदी 8 का पासवर्ड एडी पासवर्ड चली वाईफाई चली कि फोन को कनेक्ट
तो कनेक्शन कर दो ना चलनी है भीतर
मोर तो टोटली डाउनलोड ही होनी
ठीक है बाहर को पड़े जाना लेके लैपटॉप आ भीतर किसी चलनी
चालू पड़ा
आ भीतर चलो ही नहीं वाईफाई छड़ा किसी टिके ये नोटिस बोर्ड पाक सब टेक नहीं कार हमको कहता जिते सारे प्रेजेंटेशन अच्छे सब दरकार अरे सी पेन ड्राइव रे रखुंदी जो पेन ड्राइव नहीं ता आम पेन व्हाट्सएप रे पठेले पेन ड्राइव को नहीं कि से पेन ड्राइव अच्छा हाउ तू टे येटा नहीं कि तांकर कर देलु एठी नेटवर्क पाउनी एठी बाहर को जाय पड़ो
एडी वाईफाई टा कनेक्ट कर देना मैं पीजी को तापरे एटारे
पीजी को पठे दब गुआडे
व्हाट्सएप रे पठेले तापरे से एटा को नहीं जिवे एडी वाईफाई रो पासवर्ड जानो
तोर वाईफाई भी आसुनी
ना मोर तो चालू जाऊ
हाँ सेक्शन देखिलो ताके रे सब हां हां
नहीं सर
पठेची
So, welcome everybody after the tea break. So, we will be having 6 lectures before the lunch, and all they are very young professional in this field, having lot of experience in teaching as well as research. So, in this session, I request everybody to stick into the time because we are in the behind the schedule. So 5 minutes maybe the talk, 2 minutes maybe the discussion. And during the lunch time also we can have some discussions or any other queries if it is there itself. So first of all, I would like to ask Arun Rathaji, very well renowned and very good teacher, for his lecture. Arunji, please.
Good afternoon, everybody. Respected chairman, co-chairman, rapporteurs, my teachers, my friends, my head of department, हाँ ठीक है. I am Dr. Arun Kumar Raut from C. Jaydev College of Education and Technology. I like to present my original work and my personal farm, which is at Moribond district. Good. It is so everybody know about organic farming, so it is no need for discussion in details because nowadays everybody likes organics. Everybody is crying for the organics because day to day there is a major problem in the health, particularly the cancer. Then it is not to discuss because organic means that is a application of any type of plant or plant related materials or animal related materials, not toxic to the human being as well as also to the soil health. So that is the basic fundamental things, no need for discussion. So increase in population make a compulsion to stabilize the agriculture production, but to increase it further in sustainable manner. Everywhere we will get the word organic, sustainable like that. So for that purpose I have selected my own farmhouse having 8 acres of land where, sir, very less, where I am using the organic system, everything is there, vermicomposting and cows, buffaloes, and my retirement is around 2030. After retirement, my target is I have to plant 100 coconut trees, 10 cows, then 100 goats, organic, then 1000 organic hen or chicken. That is organic because they will eat all the plants, not any other ones. And I am having now 4 number of cows and milks they are producing around 10 to 12 liters per day. So these are organic because I am only taking the milk in my farm and no other, nothing where else I am not taking the milk. Only from the farm, whenever I am visiting the farm, I will take the only milk from the farm. But Savita is taking everywhere. Now whenever I am visiting my farm, only that time I am taking the milk. But that resolution I made in 2026, January. So anyhow, it is in Kadmasul, Bethanat district is a very good block because for your kind information, the Ingress, they first came to India, particularly to Bethanati. They have started the business after they visited the Balasore, then after they migrated to Calcutta because due to the mosquito problem. So this is the Bethanati. There are so many. It's the largest district in the Odisha, Moribond, and Bethanati is one of the nearer to Balasore district. So objective is to build up eco-friendly and cost-effective methods because everywhere the cost is a major problem for the farmers. For that reason, I made very simple things for the sustainability. So methodology is very simple. I have segregated the farm product and the particular kitchen product and made it to organic one or compost by using the RCC ring bin or the pot, etc. Then this is the vermicompost side at the starting. This is in my farm and azolla tank also ready for the culture. We are culturing the azolla. We are also giving to the hen, cows and goats also. Then if you go to the result is very interesting. So I have taken for the 10 days, 20 days, 30 days because I am working it here. So my people has to do it. Anyhow, they have sent the sample, etc. I analyze it, or also I am analyzing it in my own house because I am having the microscope, pH meter, etc. at my house, and balance, etc. Minimum things we are having. Sir, very less. And
why don't you have this dog farm? Sir, we'll be discussing on lunch.
Sir.
No, why head? Please care why head.
So I'll discuss with you later. Anyhow, I have considered the amaranthus because a very green leafy vegetable. Every day we are taking the sag, but you are not taking the sag really. You are taking the cancer. The sag or the green vegetable, leafy vegetable are purchasing in the market, they are full of cancer because they are using the urea and other things. Because the green means that is a cancer. For that reason, I have not purchased the sag for last 25 years. Because I have to generate in my own home at the rooftop, particular Ghatikia. Anyhow, I have done that experiment. Anyhow, finally it was observed that the organics one is better than that of growth, etc. as compared to inorganic. Then also, spinach, coriander also in my rooftop now. Also that also we have done in the field condition. Then other parameters also, that is OA means organic application, IA means inorganic application. Then finally, it was observed that organic was very good in growth, vigorous weight and other agronomic parameters, including microbiological parameters. Then this is the sweet corn. This is my own farm where we have used applied the organic one. Then you just see, this is the organic one in the very thick or their diameter is more and very higher weight as compared to inorganic one because the below one the inorganic. Then you see, the inorganic product means if you purchase the brinjal from the market, after 1 or 2 days you will get the rotting, etc. And there is no actually that stem borer or the fruit borer, but rotting, etc. because the higher application of pesticides, that is deadly pesticide we are not using, they are using farmers. 100 gram or the 100 ml is cost is around more than 2,500. So this is the brinjal. Then screening of organic crops, because I have to go for the economic way that I have screened the different crops in my farm by the application of different organic components. Then finally, I got the result. This is the zucchini, sir. Actually, my zucchini it was harvested. One was around 1.5 kg. When I sold in the Bhubaneswar market, they surprised because that is organic. They told that it is having the seed. I directly told you cut in front of me. If there will be seed, then I will take it from home, take it to my home. If not, seed is there, you have to take. Anyhow, I have win the game. Finally, that 2.5 kg zucchini, the cost is around in the market nowadays in Reliance around 300 rupees per kg only. This is the particularly guava also organic, sir, because I am actually planting the Thai variety and Thai pink, Thai 7, but it is Vietnam one because our VNR is not good. Because the pulp is not good, not tasty. We are using the Thai one because Calcutta is nearby. I purchased from the Calcutta, our train, etc. They are sending the variety, etc. I am only having 1,000 guava plantation. Then this is the totally wild one. Then this is the coconut you see. This the height is around 5 or 6 feet. The good coconut is there. There is no disease. This is the potato. This year we are doing actually. Then this is the organic oil. For a kind of information, for last 3 years or 5 years, I am not using the oil from the market. I have actually cultivate the mustard, then I actually pressed it in the Bhubaneswar, then I used. If any problem is there, you can ask to Savita because Savita is directly your student, sir. Then this is the new methodology we have implemented to actually weed problems, so that's why we are using the box type of method, and this is a very new one, and also mulching type. Everybody doing the mulching, but the farmers are not doing the box, because box you will do it, you will very easy to, it is very easy to harvest it. So box method is the mulching method. Then this is the right hand side, this is the actually red banana. That is a organic banana, or we can say it is a medicinal banana. I am having also now at home. Around 5 to 10 trees are there, banana trees are there. This is the red banana. The organic is very nice, sir. And this is the actually yellow watermelon with mulching system. And this is the directly from my farm, you see. How many number of chilies are there? Then this is totally 100% organic. This is the drumstick. You see the plant actually. How many? There is no leaf at all. Only the fruits are there. Then I focus on what? water footprint, because day to day, actually everybody is crying about the rice cultivation, government is crying, farmers are crying, but they are crying for the cancer, not for the rice, because rice actually will not get very good amount of benefit, because I am own house or the own home, we are having the 20 acres of rice field, but there is no profit at all. So anyhow, if you go for the water footprint, then there is a knowledge, a cup of coffee needs 140 liters of water. Then you see rice around 3000 liters of water, but coconut needs around 2687 liters of water per 1 kg of coconut. So highest actually beef requires around 22,000 liters of water. So to save the water, we have to transfer the rice cultivation, particularly to the other organic crops, etc. Because rabi crops actually needs rice cultivation more amount of water. That is the problem. So this is a today's cut. Government has given focus particularly on the coconut. I have discussed with my wife that she just budget actually passed for the coconut. You see, सरकार को उपकूलवर्ती अंचल में उच्च मूल्यर फसल को समर्थन करेंगे जेपरिक नडिया फर्स्ट देन चंदन. So coconut is a very good pension plan for the new days students and also for the official person. No sir, not required. Sir, I am not having saline climate because Mayurbhanj having the no saline. So I have actually calculated the so many things. Around 1.35 lakhs to 3 lakhs per acre you will get from the coconut itself. If you go for the intercrop, then you will get very good, around 5 lakhs per year per acre. So in conclusion, I can say the organic farming improves the economic status of the farmers as well as the health status of the farmer as well as the health condition of the soil also. And also it balance the microbial flora as well as the weeds also they are balancing through the organic farming. Then summary, I can say organic farming is a great alternative to conventional farming practices. And before Green Revolution, we also used the organic farming, but after Green Revolution, also we are trying for the organic farming, and definitely it is going to be good profit in a long-term basis. So thank you, sir. And this is my book, actually based on organic farming.
Thank you, Dr.
composting,
Rath.
etc.
Now
Thank you. Any question from any side?
Session is open for discussion. Any query, any questions from any side?
Sir, if we'll go for the coconut, sir, I have studied the Arabs only. Normal plating I have done, it is higher number they are coming. Population is more, sir. Yes, Arabic microorganism, because we are going for the totally Arabs, because in our, from soils of the actually, sir, rhizosphere zone of the coconut, rhizosporic soil. The Arabs, total Arabic population are higher than that of the other crops, sir. Because in coconut we are only using multiplex company Nimin, then coconut mixture. Only I prefer in the multiplex because that is a Maharashtra based company and these are organic based, sir. Mohanim, Nim, etc. we are using. Then we are using the, sir, actually cow urine. And they are increasing the population of microorganisms. Then we are using the compost, then vermicompost. Sir, whenever you are coming to the Baripada, etc., please visit my farmhouse.
Quantitatively you have studied.
Sir.
Number is increasing.
Yes. 100%, sir. And in the coconut, we are not applying any type of insecticide. Only we are applying the mustard cake, and second is your pocha, and third is the actually, sir, neem cake. Because in Mayurbhanj, the neem cake is very low cost, around 30 rupees, 25 rupees per kg we are getting.
Okay, thank you.
Thank you.
Dr.
Thank you, sir.
Rahul.
Thank you to the organizer
Now-
providing me a scope to present my not research work or my personal work,
Now,
research like that. Thank you,
I
sir.
would
Thank
like
you.
to invite Dr. Prabhat Kumar Das. Dr. Prabhat Kumar Das. Okay.
Good afternoon all. Myself Dr. Prabhat Kumar Das. Today I am explaining AI and plant taxonomy. Now this is the era of artificial intelligence and this is my topic. Will artificial intelligence replace the plant taxonomist? Is it possible? First of all, I will like to tell the plant taxonomy which deals with plant identification, classification and naming. And by morphological and anatomical also, genetic or ecological inheritance also will take as a plant taxonomist. But What is artificial intelligence shows? That is a computer system which is capable for performing any task which is such as image recognition, data analysis and discussion making, which is always help a plant taxonomist. That is the scientific background. The modern taxonomy integrates about bioinformatics, genomics and AI level based analysis. First of all, when describing a new species, we just take as a preparing flora monographs, herbarium development and specimen curation, understanding the plant evolution and biodiversity. For example, discovery of new plant species from Odisha taxonomy such as Gingiber jagannathi, Lupha egyptica variety alba, and also Abelmuscus odissi. It is the new species which is described in Odisha taxonomist. And this is the architecture of plant identification, that is by weathering, combining the image characterization, that is only leaf. We take the leaf, which it is a single leaf or compound leaf, and then considering the flowering color, varieties of flowering color, and second the fruit types and also morphology of seed types, we also segregate the plant families and directly to the plant. For example, if we consider 3 leaves and the plants having spine in the bark, then we have two, and the flower color is red. Then we have two plants in Odisha, that is Bombak Siba and one is Polaso and another is Simili. If the plants having this is the characteristic of bark, and that is the final, that is why AI and plant taxonomist. AI is calculated very fast and plant taxonomist is moderate, and speed is very fast and moderate, and accuracy is AI, for AI it is high and plant taxonomist is very high, that is expert based. For example, for Lauraceae there is particular group which working on particularly family Lauraceae, so they have the expertise in particular family. So for their data they have the expert rather than the AI. And creativity, AI have no creation, no creativity, AI have no creativity, but plant taxonomists have the creativity. They have analysis the particular data. And for field knowledge, AI have no field knowledge and no ecological data also. But for about decision making, AI have algorithm based and plant taxonomy has scientific judgment whether the plant is established or not. As a taxonomist, I also prefer sir, parameters. But sir, parameters sir distinguish करिया ପାଇଁ sir नहीं, खाली माने AI हमको help करिवो प्लांट टैक्सोनॉमी की अधिक मजबूत करिया ପାଇଁ, supporting tool. For example, we also use the as a PlantNet and iNaturalist and also Grok AI as for plant taxonomist for help. That is the integration of AI, genomic and classical taxonomy and safe future biodiversity research. That is my motto. Machines can process data, but only humans can understand the nature. Thank you.
Thank you very much. Thank you very much. Only one question. Only one question we can allow.
No, just there is a very small question, sir.
Yes, sir.
No. What kind of AI tool you want to use here? You Google Lens you are talking?
No, sir. I just talk iNaturalist.
Okay.
Then I am not.
And what will be their basic data source?
Sir, basic data by using the iNaturalist, we just observe the photographs, and in the data store they have the ecological data and also the image recognition by the leaf and etc.
Similar, if they looks very similar, you know the cryptic species?
Cryptic species, yes.
Then what you will do?
I have also mentioned, sir, there, a cryptic species.
But the slide are very beautiful. If you have made your own, it looks very beautiful. I appreciate that. It's a really commendable job. Listen, just I wanted to say one line.
Yes, sir.
Being a taxonomist, last 20 years experience says that no one can replace the classical taxonomy. Doesn't matter AI tools or
Yes,
that any other tools is coming or it will come.
Yes, sir.
Say for example, Leguminosae family.
Yes, sir.
You know, it was given by the old this classification. As of now, if we are coming to the phylogeny, they are saying that Leguminosae family,
Yes,
no 3 subfamilies are there. So likewise, many of the examples are there. So this is my personal opinion. No one can replace the classical taxonomy, but yeah, as a tool, AI is okay. That's a very good talk. Thank you.
Okay, thank you. Now I would like to invite Dr. Manas Kumar Tripathi.
Good afternoon all. I would like to thank the organizing committee to giving me an opportunity to share some of my works which I have done in University of Texas, Austin. So the title is Structural Engineering of a Nuclear Apparatus Enhancing Growth. So what is apparatus? So apparatus is basically a D-NT pages which cleave the phosphate from ATP to ADP and ADP to AMP. But in AMP, it cannot release the phosphate. So it is present in plasma membrane and also in nucleus. So what is the mechanism? Our objective when we start our work, our objective was actually apparatus are sitting in the plasma membrane and cleaving the phosphate from the ATP. So there is a mechanism that if actually it's a established mechanism that when plant and animal cells grow, they releases ATP. So the ATP was deposited in ECM. If the ECM, the ATP concentration is high, it's affect adversely to the plant growth. So it will be dwarf plant or a dwarf human beings. So what happened, the apparatus actually cleaves the ATP to balance the inside the cell cytoplasmic ATP and ECM ATP. And same time when it cleaves, it releases phosphate. That means if we grow the overexpressed apparatus transgenic crops, so that we will use less phosphate fertilizers. That is our main objective. So actually in 1999 and 2000 there was a report in Plant Physiology and Plant Cell from Stanley Jeru groups. They showed that overexpression of P apparatus enhanced the growth of Arabidopsis seedling. So to make the native apparatus a better apparatus and to patent for commercialized purpose, we have to modify the native gene, because native gene we cannot patent it. So the objective was how we will make the native gene into a better gene. So there is two techniques available. One is actually a mutation, insert of mutation to make the a better protein, and second one is it's actually gene combinations. So as at that time I was expertise in mutations, we first go in mutations. So next question is where I will mute the native gene so that it will be a better gene. So actually in the native PS-NTP9 or antipase in P, there is one calmodulin binding domain. So that actually the 1999 paper and 2000 shows that that calmodulin binding domain helps in better growth. So by bioinformatically I found that there is another stage of CAM binding domain but which is not active. So our target point is if the second calmodulin binding domain will activate, then it will may support the first one and it will be a better gene. So then we insert 2 amino acids, and then this is the bioinformatically we determine the structure, how after modifications the structure was changes. That is, PS means native gene or native protein, and DM means double mutation insert proteins. Then we actually complement the construct or gene with 4 minus yeast mutant, that is phosphate minus. There is no phosphate transporters. So we complement our gene, and here we found that this one. Actually we found that the red one, DM1, it's actually up to 72 hours, its growth is higher. First we actually test in yeast. Next one is, actually there are sometimes the reviewers comments that if you want to develop a transgenic in plant, why you are testing in yeast or a bacteria? So to complement that, plant actually have long lifetime. So for model system we develop actually hairy root system in canola, soybean and Arabidopsis. Then we transform the hairy root system, our construct, and then we will find out that the soybean, corn and canola, they have the both PS and DM have a better PI uptake, PI contents. Then we actually then transform the vector to Arabidopsis, confirm through RT-PCR, and then we actually study the relative expression levels of PS2, DM, number of silics and seed weight, all the things, and then we will find that they have the higher yield in Arabidopsis. So these are some, then the mechanism is, initially it was found or it was predicted that the apyrase, overexpression apyrase enhance the growth by activating the CAM binding domain. But in our case, DM is not showing that second calmodulin, activation of second calmodulin does not enhance the growth. Actually, then we will find out that what are happening. Then we will go several experiments and then found that. So these are all molecular studies we have done, and then we found that, to make the story short, that it actually changing in nucleus. That is, it's actually chromatin remodeling, it's happening, and that gives the overexpression of apyrase a better protein. That's our bottom line. So not activation of secondary calmodulin binding domain, but it's actually acting inside the nucleus. Thank you. So I would like to
thank you very much, Dr. Tripathi, and very nice talk. Maybe you can go for only one question.
What about the nuclear organization? You said
yes,
that is also there, na?
Okay, yes, sir.
But you didn't say anything about that?
Actually, we have done chip assays and all the things, and then we found that it's actually acting the pre-nuclei we have isolated and studied the assays and all the things, and we found that the DM, whatever it's actually after inserting the DM, the enzyme was more amphipathic. So that's why when there is chromatin making histone contact region, there is changes of binding of the protein, with binding with the protein and DNA. That was changed.
So it is only helping the genetic expression.
Yes.
You mean?
Yes.
At transcriptional presence.
Yes.
That is good. And what about chlorophyll? Chlorophyll content.
Chlorophyll actually, when we transfer to Arabidopsis, because yield is high, we saw the seed yield and all the things. So chlorophyll is high also. And after testing with all these model systems, we actually transferred the original work to soybean, and it was already tested in 3 states of USA and already commercialized, and the US patent was approved for that. So it's actually 8 years work. So in 5 minutes, it's very hard to wind up all the things.
You have the training with Professor Sapori's lab, so there is no question about that.
Okay, sir. Thank you, sir.
Thank you, Dr. Tripathi. Now I would like to invite.
Sir. Yes, yes. Okay, okay, sir.
What was the reason that you chose the hair root system
Okay, sir.
for expressing this?
Okay.
Is it a transient expression or stable expression?
It's actually a stable expression. Okay. So number one is why you will choose hair root system. What I told, our main objective is express the gene in plant. Okay. So yeast we complement the gene, and we found that it acts as a phosphate transporter. Next question is, if you study in bacteria, then the reviewers usually ask, your objective is plant. Why you are testing in yeast, bacteria? Why not in a plant model system? If you go, then next question arises was, go for Arabidopsis. Arabidopsis also 2 months and 3 months, it's a life cycle, 2 months. So we have no time for 2 months. Then we go for a hair root system, which can in 10 to 15 days, we got the tissue. Suppose we take the plant, wild type plant, then we prepare agrobacterium lysogenes, and then inject through syringe.
My question is, to be very precise, where did you integrate between the left and the right border of the tDNA
Yes.
of agrobacterium lysogenes, the RI plasmid vector which you used? Where did you insert your cassette, expression cassette? In the TL region or the TR region?
It's actually just like agrobacterium tumefaciens, it's transfer the left border, right border.
Through the triparental mating?
Yes.
Okay.
So then we select in antibiotic selections just like that. Then we grow that in MS medium.
Which antibiotic do you use?
Kanamycins.
Kanamycin. Okay. So you must have got NPT2 cassette also there.
Yes.
A selectable marker.
Yes.
Along with your expression
Yes, yes.
cassette between the bodies.
Yes.
Great.
After that we do RT-PCR
Yeah.
for confirmation. Then we take the tissue for phosphate of content and all the things.
Excellent. That's
So that
absolutely
is the
excellent.
model system.
Very good. Very good work.
Thank you,
I
sir.
compliment
Thank you.
your work.
Thank you.
Thank you very much.
Thank you, Dr. Tripathi. Now I would like to invite Dr. Dileshwar Sahu, next speaker.
Esteemed chairpersons, honorable dignitaries, research scholars, and my dear students. I am Dr. Dileshwar Sahu, assistant professor of botany from Panchayat College, Bargarh. And today I am going to deliver a talk on my work, that is the ethnic methods of detoxifying medicinal plants and ethnobotanical study or survey in Bargarh district. So as we know, we have been using medicinal plants since ancient times, but we actually most of us may not know or usually hesitate to use toxic medicinal plants. But our tribal people, they can use some toxic plants, but before using that toxic plants, they detoxify those plant parts in traditional methods. And some validation work has also been done in by different research scholars, but this is the preliminary work that I have done. I have not validated, but some validation work is already found in databases also. So this is the aim and objective, but the main objective was to screen the toxic plants and their methods of use along with the detoxification process by the people of Bargarh district. So these are the material and methods. As usual, I adopted questionnaire methods and surveyed different villages nearer to forest patches and collected the informations, mainly herbal healers. They could provide the proper information regarding the traditional methods of detoxifying medicinal plants, toxic plants. So this is the survey area, Bargarh district, that we know it is one of the 30 districts of Western Belt. Actually, it is a district found in Western Belt, and it is surrounded by various forest patches, and the main forest that we know, that is the Gandhamardan Hill. And so these are, I actually in the study, I found 19 toxic plants in their detoxification process. And these are the toxic plants, that is I have described in the table: Ebrus precatorius, Aconitum ferox, Argeria speciosa, Aristolochia indica, Calotropis procera, Cannabis sativa. And this is the table describes the plant parts and medicinal uses that actually I am not going to describe in details because there is shortage of time. So I will focus on the detoxification process. So for detoxification process, actually there are certain media. Some media are used for the detoxification process. The media may be cow urine, cow dung, warm water, cold waters, or it may be some juices of some other plants like bitter gourd juice or ginger juice or lemon juice it may be. So actually this is also an ethnic setup or traditional setup that is called Dola Yantra. It is mostly used for the detoxification of those toxic plants. So here we can see the diagram. In a pot, a particular media will be taken. That media may be kanji pani, fermented rice water, or kanji pani that we are calling, or it may be that any media that has been described in the table. And in a cotton cloth, actually that plant parts will be taken. It will be made, a pocket will be made, and it will be placed in hanging position like that over the open wide mouth of the pot, and it will be put in a stove or like that, and the steams will come out, and that plant parts will be treated with steam, or it will be also, it may be immersed inside the media also. So these are the different process. So one by one I would like to discuss. Ebrus precatorius, that we know the seed is very widely used for different purposes. So here the raw seed is actually toxic. So it is detoxified in traditional way. The outer skin of the seed is removed first, and it is crushed. Then there will be a very small thin like structure inside the two cotyledons. So that will be removed. That is the most toxic plants. Toxic parts. Then the remaining parts, they will be treated in the Dola Yantra, where the kanji pani or the fermented rice water is taken as the medium for 3 hours. Then it will be cleaned with warm water. Then it will be allowed to dry in sun. Then now the seed is ready for use. Then Aconitum ferox. This is also another toxic plant. So the rhizome is widely used. So first it will be dried in sunlight, followed by dipping in urine for 3 days. Then it will be washed with normal water. Then it will be Then it will be boiled with cow milk for 3 hours. Then it will dry, and now it is suitable for use. Then Aristolochia indica, that is the common name is the panoeri, it is called actually. The small pieces of that roots are very much useful. So here also it is treated in that Dola Yantra, where the fermented rice water is taken as the media, and it is actually kept with fermented rice water for 3 months, simply immersing. Means it is immersed inside the fermented rice water for 3 months, and at regular interval of 4 days, the fresh fermented water will be replaced. And then after it will be again treated with bitter gourd juice, and it will be rubbed like this, and now it will be dried, and now it is it is suitable for use. So another is Argeria speciosa. The seed is crushed and boiled with cow milk for 1 hour, then it will be dried in sun. Then Cannabis sativa, the seeds are boiled with cow milk for 3 hours. It is squeezed to remove water, and then it is dried. Then Datura, seeds are also dipped in cow urine and allowed to stay for 12 hours. Then the seeds are rubbed with the help of washed palm to remove the outer skin, and it is dried and ready to used. Ferula asafoetida, that we know the resin is used. So the resin is actually dried and it is fried in an iron pan with pure cow ghee under low flame until its color changes to slight red. Then the toxicity is reduced. Gloriosa superba, the rhizome pieces are put in cow urine for 7 to 8 hours, followed by drying in the shade. Jatropha corcus, it is the seed is boiled with coconut water, washing in flowing water, and it is dried. Mucuna pruriens, seed is also again, it is boiled with cow milk until the outer covering is separated, followed by sun dried. So in this way, different plants are also there. So due to shortage of time, I am not actually able to describe in details one by one. So in observations, I found actually 19 plants that belongs to 14 families, and in most of the cases, the different media are being used actually, and these are some already literature found in the databases. Existing literatures are there where they have already researched, or they have already found that there is the traditional methods of detoxification is quite scientific up to some extent. For example, conversion of some toxic anacardic acid to less toxic anacadol and lowering in the terry oil concentration was found in detoxified semicarpous anacardium seed. Similarly, the strychin content of Strychnos nox bhumica seed is reduced to 0.52% and 0.62% after treatment with cow milk and cow urine respectively. So these are some already experimented data. So this is the graphical representation of use of media. So the conclusion is that according to Ayurveda, the toxic substance has an unique property to spread the body very quickly, and if only the toxic effect is reduced or destroyed, then it can be a very gift of God, and a very novel drug can be produced or developed from toxic plants. So although some extent it has been validated, so there is need of more and more study on toxic plants so that a novel drugs can be developed. So these are the photographs of the plants, and these are the photographs taken during my survey work. And this is the paper that is published in a Springer Link journal. Actually, I have done my PhD here under the guidance of Dr. Gyan Ranjan Mohalik sir, and recently I completed my PhD here.
Thank you. Now the session is open for discussion. Any questions and query from Dr. Sahu?
हाँ लिस्ट पे जाइए आप क्योंकि मैं
Table,
just one
sir?
thing yeah this one okay here in only I have the doubt about this question second number of the species Aconitum ferox
Sir.
आपने कहाँ से कलेक्ट किया इस स्पीसीज को
Aconitum ferox, actually sir, this plant is, I have not collected, but the data is there. Yes.
Okay, so that means the secondary data you have presented.
Yes, yes.
Because this species is
Very
cannot be
rare.
found in the Odisha.
In Odisha, I have
only
not
in
found
the
actually.
Himalayan region on the top altitude. Even in the Arunachal means after the 3000 meter.
Yes, sir.
So this is what another question. You move on. एक और भी था. Okay, एक ना भांग का पाता आपने लिखा है. वो just I was discussing with sir. वो भांग cannot be nicotiana. That is the cannabis actually. Just these two things you should correct one. Okay. Otherwise it's okay. Good.
Nicotiana tobacum, the local name is called bhang in our region actually. Sir? No, local name is called bhang actually, sir. Yes, I know that is bhong. That is called in Odia actually it is called bhong or bhong like that. No, regional language may vary, sir.
Okay.
Yes. That is the local name.
Acha, okay. But overall, I would say that it's a good talk. But generally, we have never talked on that detoxification of the some toxic plants. But it's a good presentation. Okay. Thank you.
I was just wondering, what is your general message to an enlightened, scientifically quite aware audience about any method of this detoxification that you have tried yourself.
Yes, yes, sir.
More scientifically validated method and acceptable method. These are the methods wonderfully you have documented, or the traditional methods of detoxification. Using say, coconut water, cow's urine, cow's milk, whatever. Is there any scientifically more credible methods which you can suggest for detoxifying the toxic effects of these important plants to a scientifically enlightened audience in this forum?
Sir, actually this is the preliminary data that I collected from the survey work. Next work is to be done actually for scientific validation, means let's do any column chromatography or GCMS like study before detoxification traditional method and after detoxification, then analyze the data where if the toxic components is reducing or destroying or like that. In that way validation could be done, sir.
Please do extend this work.
Yes, sir.
The more scientifically.
Yes, yes, sir. I will do so. Actually.
Okay, thank you.
Okay, thank you, sir.
Next speaker, Biswajit Behra. Dr. Behra.
Thank you, sir. Good afternoon, everyone. So, as the time is short, so my talk will be based on my experimental findings. So, I'm going to talk on a comprehensive investigation of antioxidant properties of 7 edible flowers with ethnomedicinal importance in Balangir district. So, when we are in, I am from Rajendra University, Balangir, and in our locality we found several edible flowers throughout the year, like some of the flowers like these flowers are having some sort of like Adagia indica, that is the limb of Bohol, Cordia dichotoma, Musa paradica, Bahunia, variegata the Fuller, and Moringa olerifera, now it is there, Maduka longifolia, and Cucurbita maxima, and these are some of the important flowers. And I have assayed the phytochemicals present. Over 15 phytochemicals is being assayed. Actually, why I have taken this one? In my during my pre-PhD coursework, I have seen that many reviewers told that flower is a short-living organ, so you might not find many findings. From there, I just go on working. Is there any phytochemicals really found or not? And after that, I could able to find out. These are some of the testing data, and this is for Cordia dichotoma, that is Bohol Codi, and this is for Musa paradica, this is for Bahunia, this is for Moringa, this is for Maduka. And after that, these are the phytochemicals found, as well as after that I go for the antioxidant properties found in different flowers. These are the experimental results, and I found many flowers do have antioxidant properties. These are the extract results. So, these are the importance of the flowers. In my study, I have found that several phytochemicals, and in the next studies, I am going to test whether there is any antimicrobial properties or any anti-cancer properties will be the next step. So, till date, I have found only the antioxidant properties of these 7 flowers. in my work. And that's all from my side. Thank you, sir.
Okay, thank you very much. Because since it's a PhD work, so I insist all the youngers to have a question. Any question? From the young generation?
Sir, yes, this is very good. No, wait.
Yeah,
You show me that slide. Show me that slide. Yeah, second one. Rightly. Okay, fine. Now coming to the picture number 2. ये जो आपने लगाया है ना लूफा के नीचे जो लगाया है, what is the flower's name? वो second one.
Second one?
नीचे down side. No, ma'am. वो just adjacent to your hand. No.
Madhuka.
Down side. Yes.
Madhuka.
No, not Moringa. Madhuka. Okay.
Madhuka longifolia.
Now, a question is related to the Madhuka.
Yes.
You know that is the 2 variety.
Yeah,
All
yeah.
right.
Yes.
Madhuka longifolia, variety longifolia.
Sir.
Madhuka longifolia, variety latifolia. Okay. That is the 2. Madhuka indica. Sorry, not latifolia. Madhuka indica, variety latifolia. Madhuka indica, variety indica. Okay.
Yes.
अभी आपने जो इसमें लगाया है, whether this is a variety proper indica or the variety latifolia, both are the valid species.
So this is actually.
Oh, that one.
So actually, latifolia, I think.
Acha, latifolia na. Now, whenever we are adding such kind of flower, for the audience, it should, here the character is not reflecting. Okay. So this makes the ambiguity. So the presentation, okay, I am talking for the PhD student, it's okay. But just this is my suggestion. Whenever we are add such kind of flower, where there is no distinction between 2 varieties, write the name there. So that the people will understand, okay, this is the this and this is that. Okay?
Okay, sir. Thank you so much.
नहीं उनका PhD वर्क चल रहा है ना अभी चल रहा है it is continuing.
No, I am going to test all those things. Yes.
Yeah, in the next meeting
Yes,
it will be prevailed.
Yes, sir.
Okay.
Yes, sir.
Yeah. Okay.
Thank you,
Thank
sir.
you very much for the lecture. Thank you very much.
Sir.
And next is Povitra Biswal. Povitra or Prabhati or Prabhati? Prabhati Biswal. Yeah, Pratibha. Pratibha Biswal, sorry. Please come.
A very good afternoon to all. I am Pratibha Biswal, an MSc Botany student from Rajendra University, Bolangir. Yes, sir. I will be presenting my topic on air pollution tolerance index of some selected medicinally important plant species of Rajendra University campus, Bolangir. From my study, the main objective of to evaluate air pollution tolerance index in our campus. Carbon sequestration potential refers to the ability of plant absorb atmospheric carbon dioxide during photosynthesis and storage in their biomass. Chlorophyll value
chlorophyll contents refers to the photosynthetic capacity of the plant, which is reduced under under air pollution. And ascorbic acid play a crucial role as an antioxidant.
A chlorophyll data जितनी देइछो whatever you have given a computer feeding हुआ पर पूरा data रखी देइछो so 3 point point is okay point पर तीनटा disease okay ठीक है को जल्दी को
And relative water content refers to the plant
your platform both are basic
relative water content refers to the plant and higher relative water content of the plant refers to the better physiological stability of the plant during air pollution. And pH of leaf extract refers to the higher or neutral pH level of the plant can neutralize the acidic air pollutants. Sorry sir. And air pollution tolerance index is a composite index that parameters such as leaf of pH, chlorophyll content, relative water content, and ascorbic acid that parameter to use calculate the air pollution tolerance index. And apart from their environmental roles, the select. Sir?
Conclusion पढ़ा.
Yeah, पूरा ही conclusion.
हाँ. ठीक है.
इधर पूरा conclusion. From the study, it is concluded the air pollution tolerance index is a useful and simple method to evaluate the tolerance of selected medicinal plant that tolerance to air pollution.
Okay, thank you very much. Thank you very much.
No questions.
Thank
no
you very
questions.
much.
No
Well,
questions.
we have a shortage of time. We will not have the question. Okay,
Thank you, sir. Yes, sir.
Thank you very much. Thank you very much.
Next, Ritu Parna Satpati. Next speaker, Ritu Parna Satpati.
A very good afternoon to you all, respected dignitaries, teachers and my dear friends. My seminar topic is objective assessment of natural relationship of some plants, which is medicinally important and the family of Cyperaceae. Introduction, objective. Cyperaceae are graminoid. These are the grass-like family, which are sedges-like structure. It is belong to from the genus Carex and the Greek word Kyperos. Objective is the seminar objective is to contribute towards the concept of traditional taxonomy. Methodology, first we start the survey and then documentation and then the collection of the plants are done, and after that herbarium is prepared, and for identification we follow the flora of Bihar and Odisha. And then anatomical characterization is done, and then phytochemical characterization is also done. And lastly, analysis of phenogram is done. Phonetics is contribution to numerical taxonomy, and it is a great honor to Michael Adamson and Peter H.S. Smith and Robert A. Sokal. The nine genera, which are Cyperos, Picreos, Fimbristylis, Furina, Kailinga, Iliocaris, Cortisonia, and Sconioplectus and Sconioplectelia are taken from the study. These are the inflorescence characters which are studied. Cyperos has a terminal inflorescence, Picreos has anthella and glomerulus. Then all the characteristics which are studied, genus Cyperos, habitat is growth habitat and stem. Mainly Cyperos have a found in wet areas mainly. Then the ethnobotanical importance, as we know previously, K.B. Satpathy also told about the Cyperus rotundus, specifically Cyperus rotundus, which is commonly known as Mutha. It has majorly the following traditional medicines used in rural areas. And Cyperus esculentalis tubers also consumed for the, it have also ethnobotanical importance. And then we go to Picreos genus, which also habitat of marshland, and Clomset triangula. And then its ethnobotanical important also traditionally used fodder. And then the genus Fimbristylis. Fimbristylis are mainly found in wet areas, and it have triangular stem. And then it Fimbristylis mainly used as fodder and thatching material, which for building of mainly वो घर बनाने के लिए जो यूज होता है. फिर जेनस फ्यूरिना, and then ethnobotanical important of Furina, it also used as fodder and biomass. Then Kailinga genus is also comes under the operational taxonomic unit, जो 9 genus लिए हैं. फिर ethnobotanic importance of Kailinga, traditional for digestive problems, favor skin elements. Iliocaris is cosmopolitan genus, which means it have mainly majorly different characters are present. And then Iliocaris is dulcis, chestnut भी उसको बोलते हैं हम लोग. फूड और मेडिसिन में दोनों की तरह यूज होता है, और इसका एंटीऑक्सीडेंट प्रॉपर्टी भी है. फिर जेनस कॉर्टेसिना आल्सो carried out, and genus Coleoplectus also. These are the true sages. And then Sconioplectus, mat tree use हुए, basket, rope making, indigenous communities माने mainly याको use करंती. Folk medicine ले भी याको use हुए, majorly burns, wounds, skin infection जते बड़े हुए Sconioplectus को हमें use करी परिबा. Then the genus Coleoplectelia, they are thrive in moist habitats. The stem usually erect or round टिके था stem. Its ethnobotanical importance traditionally used in weaving and handicraft industries. Characters with character states, आगो हमें character तार कौन अच्छी सेटा देखो तिले. तापरे character state stem type जो दी अच्छी stem hair तार absent था कि present था.
Please continue speaking in English.
Sorry sir.
Because there are some audience among won't be able to
Foliage
understand.
leaf blades. The characters are stem type, character state, foliage leaf. If in case of foliage leaf is absent, then we code that with 0. And if blade-like lamina is present, then we code with 1. And if sheath-like present, then it is coded with 2. The phenogram is majorly concluded with, it have majorly 2 groups, major group 1 and major group 2. Thank you so much for the time.
Very nice talk. for numerical taxonomy. Very, very good. Thank you.
Next speaker, Sunil Kumar Bhoi.
A very good afternoon to you all, dear representative, dear teacher, and my dear friends. Today I am going to present a seminar on survey and ethnobotanical importance of murophytes. Murophytes, first we have to know what are murophytes. Murophytes are the plants, cryptogams, phanerogams, and all the plants that present which grows on walls, substratum or vertical surfaces in less minerals content with severe back camera. Back नहीं हो रहा. Back नहीं. हाँ mural. ताकि कैसे बे? नहीं सर back हाँ. Back take करो. Front page की. हाँ नहीं objective पे. हाँ बोल देता हूँ. हाँ बोल देता हूँ. नहीं normal हाँ. Murophytes, first we have to know the what are murophytes. Murophytes, the term murophytes come from the word murus, Latin word, which means wall. The murophytes grows on wall with less minerals and the micronutrient and macronutrient in adverse conditions. So in our research we can see that the adaptability of the murophytes, they have various morphological and physiological characteristics that may be
transition. So they put the transition on that slide. So that's no, they have put the transition on that slide. So that's what is going on.
ठीक बंद कर देना है.
चल. अरे उसमें transition डाला हुआ है.
ओहो हाँ. हाँ मैं थोड़ा. Transition. हाँ ऐसे. Several murophytes we have seen that from the Euphorbiaceae family, Acalypha indica, which is medicinally important and ethnobotanically important. We can use it as a epilepsy or having eye problem. We can use with onions and grind with them, and we can put it on the neck, on the face, so that we can cure with it. And Ageratum conizoids also used as a ethnobotanically importance plants. Alternanthera sessiles, also it is used as a leafy vegetable, also as a ethnobotanically important plant. These are some ethnobotanically important murophytes. Blumia lacera, which have anti-diarrheal activity, antipyretic activity, and Euphorbia heta, which have, we can use it eyesight problem. We can grind it and apply to the. We can use it under the eye to slow down the lesion in our eyes. Andrographic equides. It has also microbial and anti-allergic activity. With it, ethnobotanical importance. And Catharans rosians, the called as Baramasi or Sadabihari, which has anti-canceral activity. We can use we can eat its twigs with its flower for several days to cure it. These are some bioactive phytochemicals present in all the murophytes: alkaloids, flavonoids, tannins, terpenoids. Alkaloids having at least one nitrogen compound, and flavonoids have in phenolic compound which helps in the ethnobotany and for medicinal purposes used for various reasons. These. Murophytes usually grow on swells, so we have various problems with them, like impact on monuments. It grows on old buildings and various structural fences and man-made structure. So it can cause structural damage to them. Root penetration cause structural damage, moisture in wall, and accelerate weathering process. So why we have considered mural as a survey? Because there are various mural which can be ethnobotanically important. Mainly, they act as a primary colonizer for bare surface, enhance urban biodiversity, provide microhabitats, and contribute to ecological succession. So murophytes can be conserved for, we have importance to conserve murophytes for loss of traditional building, lack of awareness and neglect in biodiversity planning. These are damaging the monuments, but some cases are there where importance murophytes are there. So we have to first conserve the plant, then we think about the monuments. And future research scopes, pharmacological validation can be done, molecular stress tolerance studies can be done. Result. As I have surveyed, I have found monocots 2 and dicots 19. The rarest family I have found is Passifloraceae and most common family is Poaceae. These are done in western Odisha, Balangir district. The soil acid, mainly the Pilis, soil acid Pilis called. The species possess medicinal value, and Balangir is a place where temperature change very drastically. It may be higher temperature, sometimes it may be low, unpredictable rainfall occur. Thank you.
Okay, thank you very much. Thank you very much. Only I have one suggestion. Listen, one suggestion that the plant which is grown on the monument, same plant also found in the land. So can you compare the phytochemistry of both? There will be some changes. Okay. So that will be of interesting study because in monument whether what type of component is coming up very well. Okay. And sir has some question.
Okay. This is another one. आप अपना PPT इनका जरा PPT दिखाइएगा. This is the suggestion for you beginners.
Sir.
First thing, जब कभी आप इस लेवल पे like big platform पे जब presentation देते हो ना, don't use the animation. This looks very मतलब वो अच्छा नहीं लगता है because वो आपको खुद ही दिक्कत थोड़ा होगा, ठीक है ना? You cannot concentrate on that. This is the first thing. Second one, you are now PhD student? You are doing PhD? MSc कर रहे हैं? अच्छा, okay. ये जो botanical name आप लिखते हो ना, from where you are writing means from where you are checking the name of the plant?
हाँ, international plant name index.
Plant name ठीक है. क्या while you are making this PPT, have you checked this name there?
हाँ हाँ.
चेक किया है, ओके. आप पहला स्लाइड में जाइए, आप जहाँ स्पीसीज का नाम आपने दिखाया लिखा है, यू गो देयर. और सर पीछे सभी के लिए है दिस ऑल ऑडियंस. देखो क्या मैं देख रहा हूँ, व्हाट आई एम लुकिंग देयर. नहीं, इटालिक इज द डिफरेंट, वो दैट वो एक्सक्यूजेबल है, वो मैं उस पर नहीं जा रहा हूँ. इनको जो मैं चीज बताना चाह रहा था,
हाँ वो ऑथर साइटेशन,
ऑथर्स का नाम जब यूज करते हैं ना, दैट इज अ वेरी इंपॉर्टेंट पार्ट. ओके, यू नो दैट. हियर इन आपने क्या किया है, डी डॉन, व्हाइल यू आर राइटिंग द डी डॉन, जैसे ही डी डॉट DON डॉट, जैसे ही DON डॉट लिखते हैं, दैट मीन्स डॉन इज द इनकम्प्लीट नेम, उसका कोई और भी फॉर्म होगा, बट इट इज नॉट ट्रू. लाइकवाइज वालिक WALL डॉट, इट इज ओके, ठीक है. कुछ और भी नामों में आपने क्या किया है, वहां पे फुल स्टॉप लगा दिया है, तो इसका क्या है, वो एब्रिविएशन करेक्ट नहीं है. ओके, सो ऑलवेज चेक विद द इपनी, जो आप चेक करते हो, एंड वंस यू चेक देयर एंड यू विल करेक्ट दैट.
हाँ, करेक्ट ऑथर साइटेशन उसके बाद.
दिस इज व्हाट यू कैन डू. And all the student, those who are in the under master degree or those who are doing the research, उनके लिए तो mandatory होता. अगर आप PhD कर रहे होते तब मैं आपको उसके लिए कुछ दूसरा चीज बोलता. Since you are the master degree, उसके लिए मैं excuse कर रहा हूँ.
Thank you, sir. Thank you.
Okay, thank you.
Okay, thank you. Last speaker, Sunita Rani Mandal. Next.
Good afternoon, everyone. I am Sanjita Rani Mandal, currently pursuing Masters in Biotechnology from Odisha University of Technology and Research. So my end title was Computational Assessment of Drug Likeness, a Pharmacokinetics Profiling of Phytochemicals of Soneraceae species, a Molecular Modeling Approach. So these are the contents. This is the introduction. Due to the shortage of time, I am going to okay. So these are the plants which I have selected for my study. So this is my objective. And this is my workflows. I have going to explain this so you can get what actually I am focusing to do. So so basically my work is based on plant-derived phytochemicals for oral drug, for oral properties, or can be able for the particular disease. So for this, I have the first screening was done for toxicity analysis by using data warrior. After that, I use Swiss admin analysis for check where the compound having the pharmacokinetics properties or the drug likeness properties or not. After that, I go through the PASS analysis, which stands for predict activities substance spectrum. It is an online tool where we check the compound having the bioactivities or not. So these three screening I have done for the phytochemicals, which I have reported from literature review for the phytochemicals. After that, I go for the protein which are associated with type 2 diabetes. After selecting the proteins, I go for the Ramachandran plot to check where the protein are viable in the real life or not. These are the screening I have done for the protein and the ligand. After sir, there is some part. After the screening, I done the molecular docking using autodic vena and chimera. After the docking, I analyze the docking where the dock is suitable for the type 2 diabetes or not. Okay, so these are the, you can see I have reviewed more than 200 journals. After that, we select 69 phytochemicals and retrieve from the PubChem data for chemical characterization. And these are the table which contain the data. After that, we go for the, as I already mentioned in my workflow, we use data warrior for check the toxicity. And after using data warrior, we found 42 compounds having non-toxicity. And these are the tables. So after the data warrior, we further subject it into Swiss admin. on the basis of lipophilicity, solubility, pharmacokinetics, drug likeness, and chemical properties. So these, yes sir, so these are also used some methods, and these are the results. This is the solubility of phytochemicals, and this is the chemical property and drug likeness of the phytochemicals in the in the bottom of the two tables contain the range of the drug likeness, and the synthesis of, in the last we can see the synthesis of accessibility, which is known for the how a molecule can be synthesized for drug. So this is a radar. So basically it is a graphical presentation of the phytochemicals where the chemical can distribute more in which side. So here we can see that the red line falls within the insaturation and rather than the other. And this is a boiled egg. So it is nothing but it is a graphical representation of lipophilicity and the polarity of the molecules. So for the white region shows the gastrointestinal part, while the yellow region represents the blood barrier and brain. Okay, so these are the screening. So after all screening, I have concluded that in silico analysis help us to reduce the time, give the accurate rate where the phytochemicals have a potential to that particular disease or not. Also, it show how can it affect, or also I have concluded that it give a promising phytochemicals for choosing a disease if that's much. Due to the shortage of time, I am unable to explain. Yeah, sure, ma'am.
During the lunch time, you just ask, okay, we are shortage of time. Thank you very much. I thank all the speakers starting from the morning, because it's a very long what you call session, having I think 12 lectures. So out of them, and OBS justified the lecture from the very senior persons of the field, then after lecturer, then after PhD scholar, then after student. So they are covering more or less all the aspects. I profusely thank all the organizers of the OBS and all the participants, as well as all the what you call students, those are here. Thank you very much. And Professor Ramlal ji also, I thank you on behalf of the OBS also. Thank you very much. Thank you very much.
Now
Session is over.
Now I request Professor P.K. Chand sir to felicitate the session chair, Professor Ananta Bandhu Das sir. Anatha Bandhu Das sir. Sorry, sir. Sorry. Now I request sir to felicitate also our reporter Dr. Madhusmita Barik ma'am. We are now break for lunch. Please proceed to the dining area. We shall gather for the after the lunch. We have OBS GB meeting. I request all the live members of OBS to kindly gather at room number 5 after lunch. At 3.